0.02 and not significant. So in the literature, few studies have looked at it, but they were mostly very smaller studies. And the studies showed in the literature that BRAF mutation was associated with a poor outcome after surgery, as opposed to our study. The strength of our study is relatively a larger study with a larger number of patients, and we adjusted for and looked into multiple variables and adjusted them in a multivariate model. So in conclusion, the median overall survival was more than 2 years in BRAF mutant group, which is consistent with an overall survival benefit. The overall survival between the two groups, BRAF mutant and the wild type, did not differ in a multivariate model after adjusting for important variables. So the subset of patients with some good prognostic features might still benefit from metastasectomy. So we conclude when we're deciding metastasectomy in a metastatic colorectal cancer patient, the presence of BRAF mutation should not be an exclusion criteria as it is. These are some of the prognostic factors which may favor metastasectomy in this mutant group. I would like to acknowledge and extend our sincere thanks to all the statisticians and data managers in these databases whom we have never met, but they were very kind enough to provide the data and analyze the information. And finally, I would like to thank all the doctors who very kindly and spend their time in entering all these patients' data to these databases. Thank you.
Now open the paper for questions. I might start off, Thiru, if that's all right. Yeah. Can you clarify if all these patients had the metastasis at initial diagnosis, or was it at relapse, and does that have an impact on the outcome?
Yeah, so these are the patients we selected on diagnosis, but eventually some of the patients would have had further metastasectomies down the track. So we would expect the wild type, as I mentioned in the second slide, the wild type group has had 3 or 4 times metasurgically resected during their course, as opposed to mutant group. But still, the survival hasn't changed to compare to the mutant group, I guess.
Okay. Any other questions?
Yes. Whether this patient has also developed to secondary cancer, especially is BRAF mutated.
You mean a secondary, different
primary
cancer?
Yeah.
Well, in mutant group, no. So those incidents would have been caught during the data collection. And usually these patients do not have enough, they don't live longer to develop a secondary cancer. So far in the literature, we don't have any data to support that either. Yeah, so in our series, we didn't see any secondary cancers as such.
Somatic mutations.
That's right. So most of them are somatic mutations. So there's no sort of inherited patterns as well.
Okay, thank you.
Are there any other questions?
Sorry, can we just wait till we got the microphone? Thank you.
So just a question whether these mutations have been described in other conditions, and do you know what the prognosis for this? So similar, have you looked at anything?
Yeah, so this is not only a prognostic marker, and it's a predictive marker as well. So for example, in melanoma, the presence of BRAF mutation, it differs. In the early stage cancer, its prognosis is different as opposed to in the metastatic setting. And it is a predictive marker. So there is a targeted therapy available in melanoma for treatment of metastatic melanoma. And it can be a predictive marker in thyroid cancers as well. Some of the anaplastic carcinomas and some of the differentiated thyroid cancers have shown responses to targeted therapy. Whereas in colorectal cancer, the single agent inhibition of this mutation doesn't have any effect. So in BRAF, the colon cancer, they have clearly a bad prognosis.
Thank you.
Great. Thanks very much,
Okay.
Thiru. If there are no more questions. Our next speaker is from the Department of Hematology, Fatima Ayalil, and she's going to talk about platelet function in paroxysmal nocturnal hemoglobinuria.
Good morning, everyone. I'll be talking about my research project as a hematology fellow, platelet function in paroxysmal nocturnal hemoglobinuria. Platelet function in this fascinating disorder has not been previously comprehensively evaluated, and here we describe various novel techniques by which we analyzed this in this rare, interesting disorder. So paroxysmal nocturnal hemoglobinuria is a rare acquired clonal hematopoietic stem cell disorder first described by a German physician in the late 1800s and has intrigued the hematologists over the last century. They present with hemolytic anemia, thrombosis, and bone marrow failure. PNH is quite heterogeneous in its presentation and can present classically as intravascular hemolysis or PNH in the setting of another bone marrow disorder like aplastic anemia or myelodysplasia, or can be subclinical. Prior to the advent of monoclonal antibody eculizumab used in the treatment of paroxysmal nocturnal hemoglobinuria, the median survival of patients with PNH was only 10 years, and thrombosis accounted for about 45% of the death. And the proportion of PNH clone size has been associated with thrombotic complications. However, the mechanism of thrombosis in this condition is undetermined. We all know the role of platelets in thrombus formation, just a refresher. So when there is a vessel wall injury, platelets are there to the subendothelium via two main adhesion receptors, GP1B95 and GP6. Platelets are activated and then they aggregate via another main receptor, integrin alpha-2b beta-3. And on the surface of activated platelets, coagulation is accelerated, leading to the formation of thrombin. So we examined the role of increased incidence of thrombosis in paroxysmal nocturnal hemoglobinuria. And we tried to understand whether PNH patients have activated platelets even with a low platelet count. So 5 patients with PNH was recruited from the Canberra Hospital, and the clinical characteristics were correlated with multiple laboratory techniques, which I'll elaborate during the results discussion. As in the PNH registry, as of January this year, there are 59 patients with PNH, and we have got 5 of them in Canberra Hospital, whom we recruited. So the clinical characteristics were quite variable, with 2 patients having a history of thrombosis, and 3 of these patients were on eculizumab, and blood for these patients were collected prior to the eculizumab, and 4 of them were thrombocytopenic. So could the thrombotic complications in PNH be explained by abnormal adhesion or aggregation receptor? So we used flow cytometry to analyze the platelet surface protein levels and compared them to a healthy donor and to the same day healthy donor and a donor cohort of the ANU. So the platelet adhesion receptors, GP1B alpha and GP6, along with the aggregation receptor, GP2B beta-3, that's the same thing as integrin alpha-2b beta-3, and ADAM10, which is an enzyme expressed on the surface of platelets that cleaves GP6, was at the lower end of normal range. Could there be underlying platelet activation that's responsible for thrombosis in PNH? And P-selectin is an adhesion molecule found in the alpha granules of platelets, and when activated, they come to the surface. So there was no difference in the resting P-selectin levels between a patient and a healthy donor. And they were stimulated by various agonists. And even though there was upregulation, there was no difference between the healthy donor and the PNH, suggesting that we have not detected platelet activation in our cohort. is the endogenous platelet activation in PNH. So soluble GP6 is a marker that has been evaluated in multiple inflammatory conditions, strokes, and coronary artery disease, and have been found to be elevated in activation. So there was no, using an ELISA technique, plasma soluble GP6 was measured, and this was comparable to the same day healthy donor control. Next, we looked at the clot formation and lysis using rotational thromboelastometry, and numerically and graphically represented the data. So multiple tests were employed: extem, analogous to the extrinsic pathway of coagulation; intem, to the intrinsic pathway of coagulation; fibtem, to the fibrinogen; and aptem, looking for hyperfibrinolysis. So the graphs here show the height of a clot formed in the patients and the healthy donor via different, at 10 minutes, via multiple analysis. So the extem and the intem analysis shows that the PNH patients formed a smaller clot at 10 minutes. But when we excluded the platelet contribution to the clot, there was no difference in the height of the clot formed, suggesting that this data may be linked to the platelet count. And hyperfibrinolysis has impaired fibrinolysis has been said to be another mechanism for disordered thrombosis in these patients. And we have not observed hyperfibrinolysis in our cohort by looking at the aptem analysis. So PNH patients formed a smaller, less formed clot, often more slowly. And this was comparable with the data from patients with PNH, with thrombocytopenia. But when we looked at another parameter, extem minus the fibtem, that looks at the platelet contribution to the clot. So this is the donor cohort. This is the results we got from thrombocytopenic patients at the ANU. And when we compare our patients, these were lower than expected, implying there could be an underlying platelet dysfunction. Similarly, the clot formation patients of some of the thrombocytopenic patients were prolonged. Next, we looked at thrombus formation using a novel technique, digital holographic microscopy. So PNH and healthy donor blood was perfused over collagen at an arterial and venous shear rate. And using an in-house software, MATLAB, the thrombus height and volume were reconstructed. Thrombus stability was also assessed by flowing buffer at a higher shear rate. So there was no difference in raw data for the thrombus height and volume. But when we adjusted for the platelet count, there was an increased height in the arterial shear rate for the peak height and the volume, implying that there may be a thrombotic potential in these patients. So we have recruited 5 patients with this rare disorder in Canberra Hospital and analyzed their clinical characteristics with the laboratory techniques. So these patients had highly variable clinical characteristics, and we have not identified a unifying platelet lesion. However, DHM analysis may suggest there is increased thrombogenic potential in these patients. So mechanisms beyond platelet activation that contribute to increased thrombosis, along with the benefits of therapy on the thrombotic propensity, needs to be explored. So I would like to acknowledge everyone involved with this project, and special thanks to my supervisor, Elizabeth Gardner, and Dr. Di Rosario. Thank you.
This paper is now open for questions. Fatima, I might set you off. Sure. You mentioned the impact of the platelet count on ROTEM analysis.
Yes.
And certainly a group of your patients did have low platelet count. Do you think that impacted on any of your results, any of the other results?
Yeah, so majority of our patients were thrombocytopenic because there are various mechanisms for thrombocytopenia in PNH patients. So in the ROTEM analysis, those results we think could be the platelet count, but we don't think it has affected the flow cytometry or the soluble GP6 because we're looking at the fixed number of platelets for all these patients. Sure. And DHM analysis, we've corrected for the platelet count.
Have you considered you mentioned some of your patients have got low platelet counts. Have you considered the possibility that that could be due to formation of aggregates with leukocytes, because those aggregates aren't detected by hematology analyzers, for example.
Okay, so I don't think that has been postulated as a cause for the thrombocytopenia in these patients. So they get bone marrow failure, and the thrombocytopenia could be due to the bone marrow failure. So that's one of the mechanisms. So, I mean, something that we can look at.
Something to keep in mind.
Yeah, sure. Thank you.
Sorry,
Claire?
just in the front here. The 5 patients were on various medications. Did you see any association between the medications and any of the phenotypes? Or
Okay,
readouts?
so it's quite, so the 3 of the patients were on eculizumab, and we know that, that it decreases transfusion dependence and thrombosis in these patients. So the clinical characteristics here were, as I said, were quite variable, and the results could have been affected by the drug. So we would have to do in vitro studies with the drug on platelets and see the effect that it causes to exclude that. But these were trough levels.
Right. Thank you very much, Fatima. Our next speaker is Lucy Copeland. She's going to be talking on ROTEM. The title of her talk is ROTEM Provides Greater Insight into Bleeding Risk Than Platelet Count in Thrombocytopenic Patients.
I can thank Fatima for introducing the topic. So I'm going to be presenting some preliminary data on a project we've been undertaking to determine whether ROTEM analysis provides greater insight into bleeding risk in thrombocytopenic patients. So this project is one of the activities of the National Platelet Research and Referral Centre, which is a combined initiative between the ANU and ACT Health. So thrombocytopenia, which is defined as a platelet count less than 100, occurs frequently with the underlying causes being numerous and requiring time to delineate. So patients may present, usually to their GP, with active bleeding, or low platelet counts may be detected incidentally as part of routine care. The managing clinician's principal concern is to avoid a life-threatening bleeding episode. However, the platelet count has been shown in numerous studies to be an unreliable predictor of bleeding risk. When the platelet count is less than 20, you can imagine this bleeding risk increases substantially. However, even in this group, the risk of bleeding is unpredictable. Rotational thromboelastometry, or ROTEM, is a bedside whole blood coagulation analyzer that provides information on clot formation, strength, and lysis within minutes. So the aim of this study was to establish whether ROTEM analysis provides additional useful information to the clinician when managing patients with platelet counts less than 20. So we studied 25 thrombocytopenic episodes from 21 patients, and this group consisted of 12 females and 9 males with a mean age of 50. The platelet counts range from 0 to 17, with the normal range being 150 to 400. The presence of petechiae, mucosal bleeding, or epistaxis at the time of blood collection was considered to be active bleeding and was present in 7 of our patients, and distinct from bruising, which occurred in 1 patient. The final diagnoses in these 21 patients included 17 cases of primary ITP, 2 cases of, so ITP is immune thrombocytopenia, 2 cases of immune thrombocytopenia that was secondary to other autoimmune conditions, 1 case of acute myeloid leukemia, and 1 case of sepsis. ROTEM analysis was conducted within 4 hours after blood sample collection and following haematology consultation, and 2 of the ROTEM assays were conducted on these patients. So the XTEM assay, which examines whole blood coagulation following extrinsic pathway activation with tissue factor, and the FibTEM assay is exactly the same as the XTEM, but in addition, platelets are neutralised. So essentially in this assay, you're examining fibre and clot formation. So the relevant ROTEM terminology, I've limited to just A10 in this presentation, which represents the amplitude of the clot 10 minutes after the start of clot formation. And you can also calculate the platelet contribution to clot formation by subtracting the FibTEM A10, which is the fibre and clot, from XTEM A10, which is the whole clot, which essentially gives you the level of platelet contribution to the clot at that same time point. So these are some examples of the ROTEM output. On the left-hand side, down here we have, so this is in a healthy control who had a platelet count of 209. So in the XTEM assay down here you can see the amplitude at 10 minutes is 53 millimetres, and then the FibTEM assay the amplitude at 10 minutes is 12 millimetres. So subtracting one from the other you can see that the platelet contribution is 41 millimetres to this clot. So in our cohort of patients, we also found that platelet count is a poor predictor of bleeding risk. So I've just graphed all of the cases here against the y-axis, which is the platelet count, and I've indicated in the red circles the patients who are bleeding. In the green circle is another bleeding patient who had a very high, well, a high fibrinogen, and the case indicated in purple bruised easily. So even if we just look at the cases that are below the mean, so the bottom half of the graph here, you can see that in this cohort we have 9 non-bleeders versus 5 bleeders. So you can see that platelet count doesn't give you an indication of who's going to bleed or not. We also found that fibrinogen or the FibTEM A10 are also poor predictors of bleeding. So in 11 cases on the left here, fibrinogen was evaluated by ACT pathology, and when we correlate that with the FibTEM A10, you can see that they correlate extremely well. And when we plot all of the FibTEM A10 values for the patients and look again at those cases falling below the mean, even in this group we had equivalent numbers of bleeders and non-bleeders. So just examining the extent of fibrin clot formation will also not give you an indication of which patients are going to bleed. What we did find, however, was a more reliable predictor of bleeding risk was when you looked at the platelet contribution to clot. So as I mentioned previously, that's when you subtract the FibTEM A10 from the XTEM A10 value. So what I've done initially is I've plotted the non-bleeders onto this graph. So you can see there's a very good correlation between the platelet count and the platelet contribution to clot, which makes sense. However, when I then plot the bleeding cases onto this same plot, you can see that their platelet contribution to clot is lower than the non-bleeders, which implies that in these cases there's a certain level of platelet dysfunction occurring. What we suspect is that the autoantibody that's generated in patients with immune thrombocytopenia may be impeding the ability of platelets to function. For example, it may be binding to GP2B3A, which enables the platelet to cross-link with fibrin. And so this would result in a reduction in platelet contribution to clot. But also there may be other receptors on the platelets that are targeted by the autoantibody, and that autoantibody is impeding the platelet function. So these are some additional ROTEM examples from these cases just to show you the difference. The top two are the ones that I've talked through previously. They're from our healthy control. This middle one is from a case who had a platelet count of 6 who was not bleeding. When you look at the XTEM A10 value, it's 19 millimetres, and the FibTEM A10 value is 12 millimetres. So therefore, the platelets were contributing about 7 millimetres to the clot that formed. The bottom set of graphs, however, is also a patient with a platelet count of 6 who was bleeding. So when you look at the XTEM A10, you can see it's only 10 millimetres, and the FibTEM A10, the fibrin clot formation, is 9 millimetres. So the platelets were not contributing to this clot, which suggests in this case that there is a significant impedance of platelet function. So future work, we aim to investigate the underlying cause of this apparent platelet dysfunction by measuring the platelet surface protein levels and also identifying the platelet target that's being, the platelet protein that's being targeted by the autoantibodies in the ITP cases. So ITP is a diagnosis of exclusion and a common cause for an isolated thrombocytopenia, and bleeding risk cannot be determined with currently available laboratory tools. In our study, however, and in this patient cohort, we found that ROTEM analysis was sufficiently sensitive to detect impaired platelet function in this severe thrombocytopenia cohort, which was associated with active bleeding. And other platelet function tests remain unvalidated in such patients. The majority of them are too difficult because there's insufficient platelets to be studied. So ROTEM may assist in determining whether supplementation of specific plasma factors or tranexamic acid, for example, would enhance hemostasis in these patients. And ROTEM analysis may also provide clinicians with reassurance for patient discharge from hospital or whether these patients can undergo minor surgical procedures. So I'd just like to thank our funders, which include the ANU, ACT Health, and also the National Blood Authority. Thanks very much.
Papers open to questions. Lucy, sorry.
As a clinical test, is there a quick turnaround on that result to allow decision making?
So at the moment, unfortunately, there is only a ROTEM machine at the Canberra Hospital within theatres, I believe. We have a ROTEM machine at the ANU, so no, you can imagine the turnaround time's not great. Although we can have it, if we have a driver on site, when the blood's collected, we can have it back within 30 minutes. Yep.
There's a question at the back.
So it was sort of implied, but in your cohort you had several causes of the thrombocytopenia. Were all of the people who were bleeding, were they due to ITP? And did you see any trends with whether it was primary or secondary?
Yes, you're right. All the bleeders did occur in the immune thrombocytopenic patients. And the majority of the bleeders were newly diagnosed patients who had not received treatment. So we tend to get less bleeding events in patients who have chronic immune thrombocytopenia.
Thanks.
Lucy, have you also considered the possibility that there might be non-platelet factors that might be perhaps contributing to the non-bleeding phenotype in patients?
To the non-bleeding phenotype?
Yeah. So things like thrombophilic conditions, for example, even if they're soft risk factors.
No, so we haven't looked at that. Certainly, once you get really low platelet counts, if you also get moderately low fibrinogen, as in if you had a normal platelet count, it wouldn't have an impact, but once you get the combination of low platelets and a low fibrinogen, then you tend to get a greater risk of bleeding as well. But that's basically as far as we've taken that analysis.
And what sort of sample size do you think you need to be able to say categorically that this might be a useful test in clinical practice?
So we would like to get some more bleeding events. Fortunately for the patients, we haven't had any severe bleeders. These tend to be fairly mild cases, but we don't want to go wishing for severe bleeding cases to occur just for research purposes. We have also collected all patients up to 50, so this was basically a sub-analysis looking at the worst end of the situation. But we will be analysing our whole data set with patients up to a platelet count of 50. So, yeah.
Right. If there are no other questions, let's put our hands together for Lucy. The next speaker is Dan Tien, who has changed her topic. Can we have the slide up, please? So JNK deletion protects against hepatocarcinogenesis in phosphorus.
Morning, everyone. I'm glad to have this opportunity to represent my lab to talk about an ongoing project in our lab. So, obesity has become a worldwide serious public health problem. It can cause metabolic syndrome, type 2 diabetes, and non-alcohol fatty liver disease. And the prevalence in Australia is so high. In adults, the overweight plus obesity rate is about 70%. And the fatty liver disease affects more than 40% of Australians. Obesity can increase risk of many cancers, especially hepatocellular carcinoma, HCC, which is the focus of my topic today. So the HCC risk increased 2 times with obesity, 3 times with diabetes, and it can increase 70 to 100 times with obesity and diabetes combined in chronic viral hepatitis. It's a long and complicated process from fatty liver disease to HCC. It's the the initial fat accumulation in the liver can lead to chronic liver damage, chronic inflammation, and multiple reprogramming through various signaling pathways. With the prolonged pathology changes, this can eventually lead to fibrosis and cirrhosis that drives HCC development. At the earlier disease progression, we sometimes observe that with the right interventions, such as diet or exercise, the disease can be reduced or even reversed, but the mechanism is unclear. Considering HCC is the second most common fetal cancer worldwide and is rapidly increasing in Australia, understanding the mechanism of the disease progression would be beneficial, so we can try to reverse the disease progression to prevent HCC from happening. So one of the proposed signaling mechanisms driving fatty liver disease to HCC development is the GNK pathways. GNK1 is a powerful stress factor activated kinase in the liver. So it can be activated by various stresses that are associated with fatty liver disease, such as insulin, fatty acid, free fatty acid, cholesterol, just to name a few. Upon activation, the downstream transcription factors can have multiple functions in cell survival, proliferation, and inflammation. So there has been a lot of studies, and also our own lab's results indicate the prolonged GNK1 activation is associated with fatty liver disease progression and enhanced HCC development. So the hypothesis that GNK1 plays an essential role in the pathogenesis of fatty liver disease-associated HCC, and by inhibiting GNK1, we could possibly reduce the HCC development. We aim to determine the effect of GNK1 deletion on the HCC development in a mouse model, and to investigate the protective mechanism of GNK1 deletion. So this is the mouse model we use to resemble what is happening in the human. So this mouse, Phosphos, has a severe appetite disorder. It eats more than it eats meat and is relatively inactive. So it's both obese and diabetic. So in our study cohort, we have 4 groups. We have a normal wild type, we have the phosphorus fat mouse, we have GNK1 knockout mouse, and we have the GNK1 knockout and false double mutant mouse. So this mouse will be normal chewing water for 24 weeks, and at day 12 of age, we inject them with carcinogen. And at the end point of the experiment, the biological, metabolic, and cancer incidence readout will be carried out. So our first observation is that the green bar is the phosphorus fat mouse. It has a significantly higher body weight, a significantly higher relative liver weight and fat mass compared to wild type control, while the GNK knockout mouse significantly reduced body weight and hepatomegaly in both GNK knockout and the double mutant mouse, but can only rescue some of the double mutant mouse in terms of the relative fat mass. Furthermore, the blood metabolic readouts show that the phosphorus mouse does have a trend of increased serum triglyceride and cholesterol level, and GNK1 knockout can significantly reduce that phenotype. So we use ALT and AST as a liver damage indicator. The phosphorus mouse has a high increased ALT, and GNK1 has a trend of reduction in this GNK knockout mouse. And this disease, this lack of insignificant, we think is due to the lack of data because we only analyzed part of them. It's ongoing. Then the histology and evaluation show that only the phosphorus mouse develop fatty liver disease, as indicated as the increased steatosis score, lobular inflammation, and hepatocellular ballooning, which are all key pathological factors to evaluate fatty liver disease. Again, GNK1 knockout mouse groups doesn't develop any of these fatty liver disease. So the HCC incidence and liver nodule were measured by both HE staining and macroscopic observation. And again, phosphorus mouse is the only one has increased HCC incidence and has the highest nodule incidence, nodule number, and nodule size compared to the other groups. Again, GNK knockout mouse groups does not develop any HCC. So to begin to understand the mechanism, we studied the insulin sensitivity. So we use RPGTT, GTT to evaluate how the body metabolizes glucose. So we find a phosphorus mouse has an abnormally higher blood, circulating blood glucose, indicating this mouse cannot metabolize glucose as fast as the other groups. JNK1 knockout groups has normal glucose metabolism abilities. So meanwhile, we find a phosphorus mouse has a really high serum insulin level compared to the rest of the groups, indicating this mouse is insulin resistant. Again, JNK knockout mouse groups have significantly reduced serum insulin level compared to phosphorus. JNK1 knockout completely rescued this hyperinsulinemia phenotype. So to sum it up, we find that in phosphorus mouse, there's increased fatty liver disease and HCC development. And the JNK1 deletion can protect mouse have any fatty liver disease, reduce the HCC incidence, and improve the insulin sensitivity and some metabolic indices. So my future mechanism study will be focused on relative pathways such as JK STAT pathway, fatty acid metabolism, and oxidative stress. So our study's clinical significance is that they pointed to a potential future therapy direction. That is, we can use JNK1 inhibitor to reverse the fatty liver disease progression and to prevent people having HCC. And with that, I want to thank all the members, past and present, in our liver research group, especially my Professor Nassim Jeff and the TCH Research Office, our collaborators, and our funding body. Thank you.
Questions for Dandan? Chris, can we have the...
That's a very nice study. The weight difference is, so the JNK knockout seems to protect from the obesity. Do you have any idea of the mechanism by which it does that? Is the food intake changed or activity of the mice, or?
Yeah. So because our mouse, the diseased mouse phosphorus has high appetite, and also have metabolic disturbance. One of the hypotheses, like the prolonged JNK activation, is actually contribute to fatty acid synthesis and accumulation in the body. So, and by knockout JNK1, and then that pathway is blocked. So, and then you can't work through JNK1 to have upregulated fatty acid metabolism. Simply as that. But I'm actually working on that pathway now, and in the future I'll have a detailed mechanism answer to you. Yeah.
Do you plan to do food intake studies, or have you looked at that?
Food intake, you mean control the diet or change the diet? What do you mean?
Or measure the amount of food consumed
These
as well?
mouse are measured. The phosphorus mouse eat, I think, two times more than their mouse, the white type. So these data I didn't present them.
Yep.
So, yeah.
But with the JNK knockout, does that change
JNK1 knockout has normal appetite, but then JNK and FOS double mutant, that one also has higher, I think it's 25% higher food intake compared to the wild type. Yeah.
Okay, thank you.
Hi, just a quick question is, how long did you observe the mice before you see the nodules on the liver and to see these other changes?
So this is a really good question, because before my study, we have various time points to look at the mouse, 3 months, 6 months, 9 months, 12 months. And then for wild type, for phosphorus, it has a nodule, like a dysplasia nodule, as early as in 3 months, and at 6 months they already develop HCC. But for wild type, they does not have any of this until like later on, like 9 months, and you know. So yeah, we as early as 3 months, you can say there's a change in the phosphorus mouse. Yeah.
And the knockout mouse were watched for like 12 months as well.
Did we end to any like time? I'm just checking. I'm not sure we did the Jenkins mouse timeline study. Yeah. Bob C. It's a B6. Yeah.
Do you mind speaking into the mic?
Yeah. Do they develop? I missed your point. High levels of glucose in circulation, or they are just glucose intolerant?
They're glucose intolerant. They have an abnormal ability to process the blood glucose. But if you look at the fasting blood glucose level, there's no statistical difference between the phosphorus and the other groups.
All right, thank you.
You might have to wrap it up there. Thanks very
Thank
much,
you.
Emma. Our next speaker is Viviane Del Gingaro Augusto, and she's going to talk on is the islet beta cell hyperresponsiveness causing obesity-related type 2 diabetes and high-fat fed nod B10 phosphorus.
Yeah, I'm going to be continuing talking about the phosphorus mice, but my study, I'm going to be working on the fatty mouse, that is animal model of Ostrom syndrome. These mice, they have a spontaneous 11 base pair deletion in the exon 8 of the ALMS1 gene. This leads to dysregulated appetite. They became hyperphagic. Consequently, they develop obesity, insulin resistance, and hyperinsulinemia. But the most interesting point in these mice is that this phenotype depends on genetic background of the mice. And if you focus on the NOD-B10 mice, when female mice are fed for 24 weeks on high-fat feeding, they are susceptible to development of non-alcoholic steatohepatitis characterized by liver steatosis and hepatocyte ballooning and inflammation, and they also develop type 2 diabetes. Whereas the BOBC mice, they are protected against these pathologies. Then these differences in the mice strain makes a great opportunity for us to understand how, under obesity conditions, how NASH and type 2 diabetes develop. Then we know from the literature that chronic food sulfate, due to overnutrition and inadequate exercise, leads to the development of insulin resistance. However, we believe that if the animals, they have robust pancreatic beta cells, they are able to increase the insulin secretion in order to compensate for insulin resistance. And they will, in this way, they will maintain their normal glucose levels, and they're going to be healthy. However, if the mice, they carry susceptible pancreatic beta cells due to genetic or acquired factors, they might not be able to maintain this, to sustain this elevated insulin secretion, and eventually disturbed postprandial nutrient levels and type 2 diabetes will occur. This will lead to elevated nutrients in circulation, which will later cause abnormalities in other tissues, such as the adipose tissue and in the liver, which will become abnormal and will lead to more insulin resistance, creating a vicious cycle. Therefore, based on this, we hypothesize that pancreatic islet beta cell failure might be an upstream event in the pathogenesis of NASH. Then to better understand this, to develop this hypothesis, we performed a time course study with female white type and FOS mice from both strains, and we fed them on chow and high-fat diet from 4 to 12 weeks of age. And we follow them every fortnightly doing measurements of body weight. And as illustrated in this first graph, displayed in red and dashed lines, we can see that NOD-B10 FOS high-fat fed mice, they gain weight faster than their counterparts BOBC mice on high-fat diet. But at 12 weeks of age, both of them are equally obese. when you look at blood glucose levels, we can see that very fast after introduction of high-fat feeding, the non-B10 phosphorus mice, they develop progressive hyperglycemia, whereas the counterparts, BALB/c mice, they maintain normal glycemia during the study. And one thing that caught very much our attention was the plasma insulin levels, which after initiation of high-fat feeding, we can clearly see that the non-B10 phos high-fat fed mice, they develop profound hyperinsulinemia compared to their BALB/c counterparts. And these levels are really elevated, was 10 times higher. And this shows that they don't have defective insulin, classic signals of defective insulin secretion. And in the literature, it has been re-emerged the concept that hyperinsulinemia could be causing obesity and could be associated with adipose tissue inflammation and even leading to damage of the pancreatic beta cells. And mild suppression would ameliorate these symptoms. Then, based on this, we aimed in this study to investigate how quickly the hyperinsulinemia develops in the non-B10 phosphorus mice in response to high-fat feeding. And we want to determine if prolonged fasting, like overnight fasting, followed by refeeding, what would be the impact on the insulinemia on the 12 weeks old non-B10 phosphorus mice. So we perform a very simple experiment in order to answer our first question. We worked with 8 weeks old non-B10 and BALB/c wild type and phosphorus mice, and we placed them on 5 days of a high-fat diet challenge. We performed serial blood collections at day 0, 2, 4, and 5 for measurements of glucose and plasma insulin. And what we observed was that during this very short period of high-fat feeding, and all mice on this diet, they were able to maintain their glucose levels. But interestingly, was the non-B10 phos high-fat fed that immediately after initiation of the high-fat feeding, they developed hyperinsulinemia, indicating that this is a very fast process that occurs in these mice. Then after, we worked again with these 8 groups of mice. We fed them for 8 weeks on chow or high-fat diet. We bled them on fed state at 9 a.m. And at the end of the day, we submit them to overnight fasting, which was a 16 hours food deprivation from 5 p.m. to 9 a.m. And this period would provide them some islet beta cell rest. We bled them in the following morning during the fasting state, and immediately after we refed them for 5 days to see how a hyperinsulinemia would develop again, how quick would take for them to raise the blood levels. And we measure blood glucose, plasma insulin, C-peptide after 2 days after refeeding and after 5 days. And as we are expecting, the non-B10 phos high-fat fed mice, they were hyperglycemic after 8 weeks of high-fat feeding. And overnight fasting led to a reduction in glucose levels. But with refeeding, those levels again went back to basal concentrations. And when we look at the plasma insulin levels, we could see that although they were extremely hyperinsulinemic, the 16 hours fasting led to a profound reduction in insulin levels, and this was similar to the levels observed for the BALB/c counterparts that were not hyperglycemic, indicating here that there is one effect because they should not be reducing their insulin levels so much because they still are hyperglycemic, as you can see here, compared to the other groups. And upon refeeding, after 2 days of refeeding, they were able, the insulin levels went back to baseline levels and remain elevated. We also measured during this process C-peptide, which is secreted in equal amounts to insulin, and same patterns was observed. And when we measure the ratio between insulin and C-peptide, we could see that the not B10 phos high-fat fed mice, they have elevated ratios, and upon high fat, which reduces with fasting and again increases with refeeding, indicating that these mice, they might have defects in insulin clearance. So in summary, I have shown you that not B10 mice, when they are challenged by genetic obesity and high-fat diet, they develop profound hyperinsulinemia, non-alcoholic fatty liver disease, and type 2 diabetes. They also present elevation in insulin levels, which starts very early in time upon high-fat diet. And prolonged overnight fasting promotes a dramatic reduction in the hyperinsulinemia in this model. But despite this major reduction in hyperinsulinemia with overnight fasting, metabolic dysregulations is still present, as evidenced by persistent elevated fasting glucose. And the elevated insulin C-peptide ratio in this mouse is suggestive of reduced insulin clearance. Based on this, we concluded that islet beta cell rest with prolonged overnight fasting dramatically reverses the profound hyperinsulinemia in the not B10 phos mice. And these findings may be of relevance for dietary management of obesity-related hyperinsulinemic states and type 2 diabetes. I'd like to thank you, Professor Nolan, for the opportunity for performing this experiment, this study in his lab, and also the members of the lab. I would like to acknowledge our collaborators, Professor Jeff Ferrer and Jane Dahlstrom, for their support, and also members of the animal facility, which helped us very much during all the years that we have been performing these experiments, and the TCH Research Office and our funding bodies. Thank you very much.
I'm sorry, but we won't really have time for questions. But please do contact Vivian at the tea break if anyone wants to have a chat with her. Thanks, Vivian.
Thank you.
Our next speaker is Daniela Andrea Espinoza, and she's going to talk about the effect of clinical depression and depression comorbid with anxiety on brain structure.
Cool. Hi, everyone. Thanks for having me here today. I'm a PhD student at ANU, and I will show the effect of depression in brain structure in association with anxiety, which is a bit less clinical to what you've seen so far. So I would like to draw your attention to a couple of important statistics before we begin. So first of all is to remember that depression is the most prevalent disorder worldwide, and it does affect 4.4% of the global population. And what we've seen so far is that people living with depression have increased over time due to population growth and aging. And if we consider that this trend is expected to continue, it's quite critical to understand the effect of depression in brain structure to establish a link between brain and symptoms to hopefully in the future create or manage symptoms a bit better and improve prevention. There have been already many imaging studies looking at this, and what they found is that people with depression do have lower brain volumes compared to healthy ones, but with very few exceptions, results are quite variable and they do not agree with one another. One reason for this might be because they're not considering comorbidities, which is when two diseases, two or more, happen at the same time. And in the case of depression, it does have a high rate of comorbidity with anxiety, and some studies have found it to be as high as 50%. So maybe by considering anxiety comorbidity a bit more often, we might be able to explain some of this variability. So taking all these into consideration, the aim of our study was to conduct a systematic review of what is known about brain and depression to estimate the effect with and without anxiety comorbidity. So we did this firstly by using PRISMA guidelines, which are well-established step-by-step sort of directions on how to conduct reliable systematic review. We looked at 5 online databases up until September last year, along with manual search of some reviews and papers. We focused on the studies that included adults that were healthy or had depression, and they used an imaging technique called magnetic resonance to provide brain volumes. But other than depression or depression comorbid with anxiety, everyone was quite healthy. We used meta-analysis, which compiled the results from each study to provide a single effect. And in our case, this effect was the difference between the volumes between healthy people and people with depression, so that we could show in comparison which brain areas were higher or lower. And when we had enough information, we divided studies into those that included anxiety comorbidity and excluded anxiety comorbidity. We also looked into subgroup analysis, which go into more, say, specific types of depression, such as depression that develops late in life, which is after 50, or people going through the first episode of depression. Finally, we wanted to investigate whether other factors like age or gender could influence the effect of depression in brain structure. And what we found is that a single measure of head size, which was intracranial volume, was found to be influenced by a high number of women with depression, but no other brain region was affected. So, having said that, let's move into the interesting part, which is results. We ended up including 112 studies, and the breakdown is better seen in your diagram over here on your right, I believe. We started from close to 14,000 references all the way down to 112 here. And taken together, we included 10,845 participants, from which close to 5,000 were healthy and close to 6,000 had depression. 68% of them were women, and their mean age was 50 years. With these 112 studies, we managed to include 7 brain regions, 7 of which were subcortical, which measure areas within the brain, and 4 of which were global, which talk about more whole brain structural changes. And the results that follow are divided in 3 parts. So first of all, I'll show you the results of the effect of depression in brain structure, not considering anxiety comorbidity, which is similar to what has been shown in the literature at present. And what we found is that a region deep within the brain, here in green, called the hippocampus, was found to be 3% lower on average in people with depression compared to healthy ones. This region is quite important for storing and retrieving memories, among many other functions though. So it may have a link to memory issues as a linkage to symptoms. Another brain region that we found to be lower was the putamen, which was on average 2% lower. And this region is part of the reward system, which is linked to the joy, say, we get when we have done something well and we receive something in compensation. But it's also linked to the addictive effect of drugs and gambling. But in the context of depression, it may explain the characteristic low mood that these people experience. The last region that we found a lower effect was gray matter, which is seen over here. So basically, it just covers the outer layer of the brain over here, and it was found to be 1% lower in people with depression. And lower volumes in this particular region are linked to global cognitive problems. So, what was the difference when we looked at anxiety comorbidity and first of all the effect of depression with no comorbidity? The effects were larger, so namely from the hippocampus we went from 3 to 4%, in the putamen from 2 to 4%, and in grey matter volume from 1 to close to 2%. So all these effects were larger. But we also found 2 other regions that had lower volumes. One was the globus pallidus and the thalamus, which on average had 3% lower volume in people with depression. So the globus pallidus here sits very close to the putamen and is also part of the reward system. So it may have similar links to symptoms. But the thalamus sits a bit more central in the brain, and some of its functions are to regulate like overall bodily functions, such as regulating appetite or sleep patterns. So insomnia that sometimes people with depression experience, or changes in appetite, may be explained by changes in this particular brain region. And what happened when we had a look at depression with comorbidity? The results were quite different. Instead of the lower volumes that I've been showing you so far, we found that this region called the amygdala could have as high as 4% higher volume in people with depression compared to healthy ones. This region is normally seen as the emotional center of the brain because it's linked with the expression of emotions such as fear, but it's also quite consistently linked to anxiety, so it may very well explain the symptoms in people with comorbidity. But when we had a look at all the brain regions with and without anxiety comorbidity, what we found is that anxiety masked the effects of depression by around 3%, which is seen a bit better in this diagram over here. On the far right, you can see the effects of depression comorbid with anxiety and the effects of depression alone with no comorbidity, and the differences are quite striking. So last part talks a bit more detail into subgroup analysis, and we found consistent results with depression in late onset and multiple episodes. And what we found is that the volumes were even lower that I have shown you so far, and this was particularly the case in the hippocampus, where volumes were to 3 to 4% to what I've shown you before, but they could get as high as 7%. So it's quite a fair bit of differences. So take-home messages from here. What we found is that the effect of depression on brain structure can vary due to anxiety comorbidity, number of episodes, and time of onset. But this later point may also might be related to aging, because as we grow older, our brains naturally shrink. So it might be a compound effect between depression and aging. And some implications for researchers and clinicians in general is to consider that maybe what we know about depression so far, how it affects brain volumes, has actually been underestimated because anxiety comorbidity has not been consistently assessed. And early diagnosis of anxiety symptoms and to continue monitoring of these symptoms might lead to more effective treatment options for these patients. And on that note, I want to thank you for listening. Cheers.
Thanks very much, Daniela. Unfortunately, we are going to have to curb questions. Thank you once again. Our next speaker is Charmaine Simenovic, who's going to talk about circulating platelet neutrophil aggregates as a biomarker in type 1 diabetes.
Thank you. Thank you very much. Type 1 diabetes is a T-cell mediated autoimmune disease whereby progressive inflammation of the islets of Langerhans in the pancreas leads to the specific destruction of insulin-producing islet beta cells. Multiple destructive mechanisms contribute to this, and a number of different immune cell populations are involved, including T cells and macrophages. But also, there's now a growing evidence for neutrophils in this process. Now, when neutrophils become activated, they release their nuclear DNA into the extracellular space as these extracellular traps or nets. And these web-like structures, shown here by cytox screen staining, it's difficult to see on the screen, are very effective in limiting the spread of infections through the body. However, in the absence of microbial stimulation, activated platelets can activate neutrophils to release nets. Here, the neutrophils recognize P-selectin or HMGP1 release from the activated platelets, and either pathway leads to the citrullination of nuclear histones, decondensation of the nuclear DNA, and the expulsion of these nets outside the cells. Now, in addition to the nuclear DNA, these traps also contain nuclear histones, nuclear proteins such as citrulinated histones, and also enzymes derived from the cytoplasmic granules such as myeloperoxidase. These citrulinated histones can activate platelets, and these activated platelets can activate the neutrophils to release the nets, and so a vicious cycle of platelet neutrophil activation is generated. Just as an aside, these citrulinated histones can inadvertently damage host tissues. So there's a very close association between platelets and neutrophils, and these interactions are known to contribute to various vasculopathies and inflammatory disorders, including autoimmune diseases. In the case of type 1 diabetes, circulating neutrophils are actually decreased to about 75% of healthy controls in children at T1D onset. And there is also a significant reduction in circulating neutrophils in autoantibody positive relatives. There's also a significant increase in circulating nets, a significant increase in the activity of neutrophil enzymes in the circulation. There's also evidence of activated platelets in the circulation. And in the pancreas, there's an increase in MPO positive neutrophils in pancreas from new onset T1D donors and also donors that were autoantibody positive. So the aims of our study were to determine why circulating neutrophils decline in human T1D and whether these changes in peripheral blood neutrophils correlate with T1D-related islet pathology and beta cell damage. So for the first aim, we analysed blood samples for platelet-neutrophil interactions from children 5 to 16 years of age with T1D or who were at risk of developing T1D and who were autoantibody positive, those that were autoantibody negative, and we compared these to healthy control children. In the second aim, we made use of our non-obese diabetic mouse model of spontaneous autoimmune type 1 diabetes, and we analysed platelet-neutrophil interactions in the blood and islets of these mice at various stages of T1D development, and we also investigated localization of products of activated neutrophils in the pancreas of these mice. Now, to investigate platelet-neutrophil interactions, we identified the formation of platelet-neutrophil aggregates. And we did this by flow cytometry, and we used CD15, CD16 to identify neutrophils, and then we looked for those neutrophils that carried the platelet-specific marker CD41 to identify the platelet-neutrophil aggregates. We found that at T1D onset, there was a 2.8-fold increase in circulating PNAs. Compared to the pediatric healthy controls, these T1D children were actually analysed by about 10 days after clinical diagnosis. We also saw a significant 2.1-fold increase in PNAs in the circulation of the autoantibody positive children. And then after T1D onset, these PNA levels declined rapidly. Carrying out linear regression analysis, there was we saw an indication, a suggestion of a negative correlation between neutrophil numbers in the circulation versus the platelet neutrophil aggregates, suggesting that this decline in neutrophil numbers at T1D onset in humans could be due to the formation of these platelet neutrophil aggregates that are not detected by hematology analyzers. We saw no correlation between platelets and PNAs in the circulation. We did, however, see direct evidence for these PNAs using flow cytometry and image stream analysis. Here we see yellow fluorescent platelets adhered to human neutrophils. In the case of our NOD mouse studies, similar to our human studies, we saw this progressive increase in platelet neutrophil aggregates in the circulation. At 4 weeks of age, we saw a 1.7-fold increase over non-autoimmune controls, and this correlates to the initiation of T1D autoimmunity. Thereafter, at 10 to 12 weeks of age, we saw a 2.1-fold increase in the PNAs in the circulation, and this correlates to when there is a major influx into the islets of leukocytes to destroy the beta cells. And at T1D onset, a 2.5-fold increase in the PNA levels. Now, in correlating with these changes in the PNA levels in the circulation, we also saw changes in the PNA levels in the islets of NOD mice at different ages. At 4 weeks, an increase in the islet PNAs here, similar to what we saw in the circulation, and then again from 10 to 12 weeks of age, a similar elevation. However, here in the islets, those levels remained elevated. These elevated PNA levels in the islets of the NOD mice correlated with histological evidence of damage in the islets and histological evidence of destructive insulitis at 4 weeks and from 10 to 12 weeks of age through to T1D onset. The platelets in the PNAs are actually activated. We found that P-selectin, the levels of P-selectin, an activation marker of platelets, were over 100-fold higher than that in resting platelets. Using immunofluorescence microscopy, we found evidence for netting neutrophils that were MPO positive, citrullinated histones and citrullinated histones with extracellular MPO in these NOD pancreases at different stages of T1D development. We found that the proportion of islets that were MPO positive or that were positive for the citrullinated histones significantly increased during T1D development, suggesting that these net products could be contributing to beta cell damage during development of T1D. So in summary, elevated PNAs may account for the mild neutropenia reported for T1D onset in humans, and we know that activated platelets produce a chemokine called CXCL7 that promotes neutrophil migration, so we think that these PNAs may facilitate the migration of the neutrophils to the pancreas. In our NOD studies, we found that these circulating PNAs represented a dynamic peripheral biomarker of beta cell damage and T1D development, and certainly we have suggestive evidence that net products contribute to beta cell damage. I'd like to acknowledge everyone who's contributed to this work and to our funding agencies, ARC, JDRF, and the Australian Type 1 Diabetes Clinical Research Network. Thank you.
We have time for one question from the audience.
Fascinating, really nice data. At the same time that type 1 diabetes develops, you get hyperglycemia as well. Are you able to control for hyperglycemia?
For the
whether
development
that
of the
affects
PNAs?
Whether that affects PNAs, so in non-type 1 diabetes, for example?
Well, we actually see, we haven't looked in, well, we look in our healthy control, in terms of our human studies,
Yep.
clinical studies, we see those elevations even in the autoantibody positive pre-T1D children,
Yep.
so that's before hyperglycemia.
Yes, so that's important. Yeah.
So, and in our mouse studies, those elevations occurring before hyperglycemia and the onset of type 1 diabetes.
Yeah.
So,
So that's
it's
good
not
evidence.
essential,
Yep.
the hyperglycemic state is not essential, but it may actually exacerbate.
Yeah, because after 1 year, they dramatically drop.
Yes,
yeah.
They dropped rapidly, yes.
Yeah, okay,
Yeah.
thanks.
Thanks very much, Emma. Our next speaker is Jessica Oh, and she's going to talk, the title of her presentation is Investigating Spatial Convergence of Diagnosed Dementia, Depression, and Type 2 Diabetes Prevalence.
All right, good morning everyone. My name is Jess, and as mentioned, I'll be talking about the spatial convergence of diagnosed type 2 diabetes, dementia, and depression prevalence in West Adelaide. So quite a general outline, I won't go into much depth. Dementia affects 50 million people worldwide, and there's serious financial repercussions with an estimated $1.4 trillion spent last year. It's the most common chronic disease in Australian aged care facilities, and studies have demonstrated that both depression and type 2 diabetes are independent risk factors for dementia, and other studies have also demonstrated a strong association between the two, which led to the research of Caton et al., where they found in their study that individuals were 4 times more likely to develop dementia if comorbid with type 2 diabetes and depression. So we thought, how about we map the prevalence of all three of these diseases and see if we can find areas of convergence, and hopefully with those areas we can have practical implications in policy. So we had three aims. Firstly, to determine the prevalence of each disease and associations with various covariates. Secondly, to determine the geographic variation. And finally, to identify hotspot and cold spot regions of convergence, which I'll elaborate on a bit more. So we used general practice data from 16 clinics, and as you can see there, we started with quite a large data set. We then narrowed it down to active patients, and then we further narrowed it down to patients that were over 45 years because of dementia's association with age. We then further narrowed it down to individuals with a reported sex, and that still left us with quite a large data set of over 40,000 individuals. We looked at each of the three diseases as their own outcomes, and we also looked at various socio-demographic, health and comorbidity covariates as listed there. So we had statistical analyses, so pretty standard individual level descriptive statistics, followed by multi-level modeling, nesting by statistical area 1 codes, which are unique codes assigned to every suburb generated by the Australian Bureau of Statistics. We then continued with spatial analyses, so we had a distribution analysis followed by cluster analysis. So in our cluster analysis, we were looking for hotspots. These are regions of significantly higher prevalence of the disease, or cold spots, regions of significantly lower prevalence. So in terms of our findings, our descriptive table here, as you can see there in the little blue box, 28.9% of dementia patients were comorbid with depression. Just under 1 in 5 were comorbid with type 2 diabetes, and 7% were trimorbid. And it's important to note here that the percentage of Aboriginal and Torres Strait Islanders was not representative in our sample, so we didn't continue with analyses using that aspect of the data. So moving on to our multi-level analyses. I do apologize for this giant table, but we'll try and break it down a bit. So we've got each disease outcome in the 3 columns. And the significant covariate across all 3 diseases was comorbidity, as you can see in the blue there. And between dementia and type 2 diabetes, the shared covariate was elderly age, as you can see there. And depression also has a strong association with age, but in the opposite direction. Between depression and type 2 diabetes, we had having hyperlipidemia, having hypertension, being a current or ex-smoker, and being of lower socioeconomic status were all shared significant covariates. So moving on to our geospatial analysis. So as you can see here, we've got the 3 diseases in their own separate maps there. And the color gradient represents differences in prevalence within the suburb. So a darker color represents a higher prevalence of the disease. And for dementia and type 2 diabetes, we had up to 31% prevalence in some suburbs, and for depression, up to 54%. So we then proceeded on with our hotspot and cold spot analysis. And as you can see here in the red, we were able to observe hotspots across all 3 diseases. And they all kind of tend to be in the southeastern perimeter of the area. And we also have cold spots, so these are regions of significantly lower prevalence. And as you can see, for all 3 diseases, they tend to be along the coastline. And that may very well be a reflection of higher socioeconomic status or access to better lifestyle habits like walking along the beach. So we then put all 3 disease prevalences together and ran the analysis again to see if we could find these hotspots, and we were able to. So we found 33 hotspot regions, again they're in the red, and again sort of in the southeastern area, and we found 48 cold spot regions all along the coastline. So in terms of discussion, most of the covariates in our statistical analyses were consistent with current literature, except for BMI and dementia. So in our study, we found that being obese was protective for dementia, and a potential explanation for that is reverse causation, where it said that weight loss is associated with dementia, making it seem as though individuals of a higher weight, so a higher BMI or being obese, are more likely to have dementia. But again, these are just postulative theories. Now, there is also a potential biological connection between the three diseases. Now, I'm not an endocrinologist, but from what I've read, there's a suggestion that dysregulation of the hypothalamic pituitary adrenal axis is associated with dementia, and that itself can cause excess cortisol, which is associated with type 2 diabetes and depression. So in terms of the strengths of our studies, we use neighborhood level analysis, and that provides a lot more detail to have hopefully more practical policy outcomes. We used GP data, which is large scale, as I mentioned. Even after narrowing it down, we had over 40,000 individuals. And there's obviously less self-report bias because you need a clinician to diagnose you with the disease. But there are obvious limitations as well. We were unable to assess some covariates just because there wasn't enough data reported. There's also a potential bias towards individuals who are more health conscious and actively visit a general practitioner. And while we like to think that the data is representative, and we hope it is been recognized in this study that 85% of Australians visit a GP, so we're sort of limited in that sense. So moving forward, I think it would be good to encourage more standardized recording of patient data. As I hope we've demonstrated, general practice data is a very useful source of information, but the veracity of the results produced from these analyses are limited by what is recorded. It would be good to include a longitudinal component to see if there's changes or new diagnoses, and most importantly, to characterize these hotspots. So it's one thing to know that there's higher prevalence in some areas, but why is that so, and what can we do about it? So in summary, we've observed convergence of these three diseases in West Adelaide, and our statistical analyses demonstrates that there are shared associations between these three diseases, so perhaps we should be targeting prevention of these three diseases collectively. And in our geospatial analyses, we're able to identify regions of higher prevalence and lower prevalence, and hopefully that can have practical purposes in targeting prevention measures for efficient resource distribution. And I'd just like to thank the 16 general practices for supplying data. Thank you very much.
Thank you very much, Jess. We won't have time for questions, so please contact Jess at morning tea if required. Our next presentation is from Sally Hall, who will be presenting a systematic review of the literature on factors associated with clinical practice variation in GP management of diabetes and coronary heart disease.
Thank you and good morning. I'm going to continue taking you out of the lab and into primary care and the world of health services research. Jessica's presentation, I guess, is a nice segue into this idea of variation and our increasing ability to measure it in practice. And clinical practice variation is an idea that is occupying a lot of our attention in the healthcare quality and performance space these days. And there are a number of definitions, but basically it's the idea that, patients who essentially look the same and seem like they should be treated the same often aren't, and that might set some red flags for problem areas that we might want to look at. But at the same time, we know that there are times when variation might be warranted and that there might be a range of reasons why that's actually appropriate. So actually what we need to try and do is focus on the idea of unwarranted variation and work out how we can tease that out. So generally, unwarranted variation, and again there are a number of working definitions for this concept, but it's generally understood to be variation that we can observe that is not explained by differences in patient needs or preferences, and therefore as a corollary is something that's likely to be explained by differences in health system performance. But essentially one of the ways we tend to try and unpack that and think about the concept of unwarranted variation is to think about sources or causal explanations, and increasingly we know that they are complicated, and that's particularly true in the world of primary care where we were working. So taking all of that on board, we wanted to
or we were planning to do an analysis of some general practice routinely collected clinical data that looked at variation in management of chronic disease. So we wanted to know what the literature tells us about the explanatory models and factors that might be associated with the relevant kinds of variation. And so we decided to do a systematic review that might inform the conceptual model we'd use in that statistical analysis. And we were looking at this from a social ecological systems kind of framework. So our review questions were these. We wanted specifically to know what kinds of explanatory factors or models had been identified, to what extent was observed variation explained by those models, and how did the factors differ between processes and outcomes of care. So essentially this was our search strategy. We looked for empirical studies over the last 25 years in the international literature. We used a 3-stage iterative process, so we scoped the literature to refine our terms. We ended up using 4 electronic online databases and 3 grey literature repositories. And you can see there the criteria for our search. So we were looking at diabetes and coronary heart disease in primary care, dealing with issues around clinical and medical practice variation. We used the PRISMA guidelines to structure our search, and you can see there that we did all of the usual things. I won't go into detail. But the key takeaway, I guess, from this slide is the bit in the green circle down the bottom, that we had 85 included studies for which we did a narrative synthesis. But a large number of the works that we found and needed to include were in fact theses, so they were much longer than a paper, because a lot of these are large database studies. In terms of study origin and design, you can see that 2/3 of our studies came from America, a smaller group from the UK, and then a smattering of papers that were spread internationally with 6 countries, with 1 study each. And that largely reflects, I think, the capacity so far for this kind of data collection and analysis, which tends to be concentrated in the USA, and to a lesser extent in Great Britain. In terms of study designs, the vast majority again were cross-sectional and observational, with a smaller group of longitudinal studies and then only a smattering of experimental study designs in this field. Breaking them down by disease, around 63% of the studies that we looked at dealt with diabetes. If you look at all of the other or the next 3 bars taken together, coronary heart disease and hypertension and hyperlipidemia, which function as risk factors for that, essentially constituted around about the same number of studies. And some studies obviously assessed more than 1 disease, which is why those percentages add up to more than 100. In terms of outcome variables that were represented in the studies that we looked at, for diabetes you can see that there were around 18 process measures that usually dealt with what are considered to be in primary care relatively standard measures of quality. So looking at whether glycemic control had been assessed and recorded, the same for blood pressure, lipid profile, nephropathy, retinopathy and so on. There were some around prescribing behaviour by drug class, or looking at preventative vaccinations, smoking cessation, and then some things around the way that treatment decisions were made. Around about 6 outcome indicators looking at glycemic, lipid and blood pressure control, and then some variations in things like how prevalence was being assessed and recorded, and then a small group of studies looking at quality of life. And then another small group that look at composite indicators, which often are process and outcome variables lumped together, and these tend to be reasonably arbitrary and very study specific. The bars at the side there are just there to show you that there is huge variation in terms of the number of studies that address each of these indicators. So looking at the top bar, top graph up here, you can see that this first bar with around about 18 studies is studies that included a process measure around glycemic control, ranging down to only 1 study looking at diagnostic accuracy. So that idea that there is huge variation in the numbers of studies that look at particular things will become important shortly. Likewise, for coronary heart disease, you can see that there's a similar spread in terms of the indicators or measures that were used in these studies. Around about 19 looking at processes that were very similar in nature to the ones that were represented for diabetes. Around half a dozen looking at outcomes, and a small group of composite indicators as well. In terms of our results, overall practice variation was generally reasonably poorly explained by the studies that we looked at, but there was huge variability in terms of the model fit statistics that were used in the way that models were reported. R-squared values or other measures of model fit were often very low, but that tended to deal with the fact that most of these studies are looking at testing specific hypotheses rather than looking at overall explanations for the variable that's been observed, which is kind of an epistemic difference in the way that studies have approached this issue and are starting to try and think about it from a complexity perspective. But it raises the question that lots of these models are likely to be incomplete. We had hoped in extracting value from this literature, which is very diverse, that we might be able to distinguish definitive explanatory kinds of frameworks for individual variables, but that wasn't possible, partly because of difficulties with study comparison and the fact that, as I showed you in the last slide, that high frequency variables tended to be associated with many more factors simply because there were many more studies. And then there was quite a lot of overlap of factors between variables as well. In addition to that, we found quite a number of contradictory effects, even for related variables. So the kinds of things that we would observe had different magnitudes and moved in different directions depending on the study that we looked at. And that raises an interesting set of implications for composite variables and the way they're put together, and to what extent they hide meaningful information. The last thing that we found overall from the literature was that processes and outcomes may behave in different ways, that while they often correlate, they don't always correlate, and when they don't, they seem to be potentially interesting and intriguing relationships between them that might be non-linear, which suggests that quality might also not be a single construct in this space. So while we weren't able to come up with frameworks for individual variables, what we were able to do was come up with an overall or an overarching explanatory framework for how we might proceed with our statistical analysis. And essentially what we identified is a range of factors that are likely to be influencing variation at 4 different levels. And you can see those there. So in terms of the environment or system level, we found about 36 factors which were clustered into 6 themes around measurement, locality, population characteristics, workforce, policy interventions, and then a number of interaction effects which show up at each level. For practice level variables, you can see many more, around 9 themes. So they're things like scale or size, staffing, practice type, revenue stream, maturity, culture, organisation of care, integration, measurement-related factors and interaction effects again. Provider level factors clustered into 5 themes, which you can see there. And then patient level factors were by far the most numerate, and we had 8 themes there. So this, in line with previous presentations this morning, talks about comorbidity, disease severity, socio-demographic characteristics, treatment modalities, service utilisation patterns, the way that patients are perceived by providers, and psychosocial and complexity factors. Obviously, there are a number of limitations, so study comparability was a huge one, along with the quality of reporting, and there were quite marked differences in context and in the way problems were framed, especially between countries, which made comparison quite difficult. But one of the other issues that came out was that there's the possibility of almost a research lens bias in this work, in that there are many studies who all look at the same things, but few studies that try to approach this in different ways, which raises the question of whether we've actually been looking under the lamppost and provides food for thought. So finally, our conclusions at this point. Explanations for clinical practice variation in this setting are likely to be complex, multifactorial, contextualised and interactional. Warranted variation is often conceptualised in terms of causes or sources, and our framework offers a theoretical structure for exploring and clarifying those relationships as a way of unpacking what might be warranted and what's not. So thank you for your attention.
Thanks very much, Sally. We won't be able to take questions for this one. After the last two excellent clinical general practice presentations, we have James O'Connor now, who's going to take us back to the lab with his talk on intravital imaging in detection of antibodies in Plasmodium falciparum.
All right. So like Dipti said, I'm going to take it like Sally's taken you back out of the lab. I'm going to bring you back in, which is, in my opinion, one of the better places to be, but that's my biased opinion. So my talk's going to be on intravital imaging and how it's revealing anti-parasite activities of antibodies targeting the Plasmodium falciparum CSP protein. So what is malaria? Most of you have heard of it. It is caused by the parasite Plasmodium. It's an obligate eukaryotic parasite. It affects a lot of African nations as well as South America, Southeast Asia, causes anemia, headache, diarrhea, and in severe cases, death. Now, the incidence is rising with 219 million new cases. And if any of you are from Adelaide, I'm sure that you probably will be aware of the latest outbreak of malaria that they had, where there was about 6 different cases. So while it might not be a cause for alarm, it's not too far from home. So this is kind of one of the reasons that keeps driving me forward, is it's not just a third world disease. It is a pretty significant one at that. So what about the life cycle of the parasite? I'm not going to bore you with the nitty-gritty details about it, but I just want to give you a quick overview of the fact that it exists both in the mosquito and in the mammalian body. And you can see that it starts off as a sporozoite, as a parasite sitting within the mosquito, travels through the blood from an insect bite to the liver, where it infects the liver cells or the hepatocytes. From here, it develops into merozoites and becomes what we call blood stage infection, where it bursts out of the cell and begins to infect the red blood cells. This is where it becomes symptomatic, and this is where a lot of drug targets are sort of focusing their work on. But what I like to really focus on is the fact that you have a bottleneck here. This is the stage that you can really stop it. So if you can get the stage where the sporozoite becomes a merozoite in the hepatocyte and stop it from actually getting out and developing blood stage infection, this is going to be a really critical time where you can target it. So I'm going to focus a lot of my talk, and this is where a lot of my research tends to focus on, stopping the actual infection from sporozoites in the skin to the liver and then bursting out. So how do you actually do it? CD8 T cells have been shown to actually cause sterilizing immunity in these models. So if you have enough CD8 T cells that are specific for hepatocytes that are infected, they will kill all of the parasites and you won't get blood stage infection. But it's not very practical unless you have CAR T cells or if you adoptively transfer all these activated specific cells. So the other alternative is antibodies. And antibodies are really sort of the foundation of these immunotherapies that you tend to see in a majority of treatments at the moment. And this is probably the most promising that we've got so far. So my project looked a lot at CD8 T cell homing and migration to the liver, and they do a good job, and you can't do it without them. But if we can actually get antibodies that are targeted to the specific proteins on the surface of these parasites, it might actually be a really good immunization strategy to protect. So the 2 main questions that you sort of have are, okay, what defines a good immune response? You need good quality specific antibodies, and you also need protective CD8 T cell responses. But then what are the characteristics of these factors? What are the characteristics of a protective antibody response? And what defines a protective CD8 T cell response? I'm going to focus on the former and not really talk so much about the latter in these situations, but the whole project looked at the holistic view of how these 2 mechanisms actually control infection. And for the purposes of today's talk, it's going to be antibody-mediated protection. So back in the 90s, they had a look at sporozoites in cultures, and they took serum from people that had been essentially immune to malaria. And what they found was that when you treated these sporozoites with the serum, they tend to ruffle up. The skin rolls up, they don't look very good, they don't move very much, and they tend to die. And the reason for this is that in the serum you had antibodies that bound to this repeat region that you see on the protein that's expressed on the surface of a sporozoite. So here you've got a sporozoite, here, again here. When you add the serum, they tend to not look very good. And these antibodies tended to bind to this one here, this region of the protein. So how did that actually look in a schematic view? You can see on the top here you've got the normal structure of the protein. I'm not going to bore you with the specifics of this, but essentially the RTSS vaccine, or the current one that they're trialling in Africa to protect children from dying, is using this one here. It's a truncated form of the protein. They've added an adjuvant to it, which is a hepatitis B monomer, antigen monomer, and they use just a small region of this repeat. Is it any good? Not really. It's better than nothing, but what you can see, they've compared it in a few different slides. There was a paper in New England Journal in 2013 that sort of showed that it was slightly better than the rabies vaccine, but it didn't last very long. And again, you can see in these studies here with African children, you find that even after 3 boosts of the vaccine, you tend to not get a very good antibody titer in these individuals, and it's not very protective. And even if it is, it doesn't last very long. So what does that really mean? Well, recently what a study in Nature Medicine found was if you actually inject humans with the whole sporozoite and you irradiate it, so you've got the whole protein there, the immune system is able to recognize it, generate B cells. What they did was when they immunized these individuals with these radiation-attenuated sporozoites of falciparum, they call it Cenario, which is a trademarked drug, or a vaccine, sorry. They took the single cells that were specific for the CSP antigen, or the NANP. They identified these specific clones of B cells. They then took the antibodies and they found 2 that were very, very good at stopping infection. And for the purposes of this talk, I'm just going to call them MAB10 and CIS43. And they found that these antibodies bound to specific regions of the protein. You've got the region here that sort of borders between the N-terminal domain and the repeat region, and then just MAB10, which sort of sits on the repeat region. When you use these in mice, you find that when you use it at 150 micrograms and then infect them with malaria, you have a almost 10-fold decrease in liver burden. And even when you take it down to 30 micrograms, you still have roughly the same amount of protection. In blood stage parasite studies, where they did long-term ones, if they inject these mice with these antibodies, they're actually really quite protective, CIS43 much more so than MAB10. So the question then we asked is there actually any potential to use these antibodies synergistically? Because they're both very good on their own. And how do they actually work? How are they stopping infection from occurring? And this is where we use 2-photon in vivo microscopy. So this is a technique that we use in our lab that actually lets you see what happens in real time in a real live mouse with a real live parasite, what's actually happening. Now, it's important to just quickly run over the actual microenvironment and cellular composition of the liver. You've got these sinusoids, which are just single-celled endothelial vessels that run through the liver, and you have hepatocytes that sit in the center. And the parasites tend to come in through the blood, and then they infect these hepatocytes here, where they tend to cross through a few, then they establish active infection, and then go on to produce blood stage. Now, what we've been able to do is use a few different imaging technologies which we thought were pretty cool. So initially we wanted to look at what platelets were doing in the liver, and so what you can see is you've got red that sort of symbolizes the hepatocytes, and then you've got these platelets that are just flowing through. Sometimes they bind to the endothelium, sometimes they don't, and they just move through. This is in the absence of infection, but that was a different arm of the project. We then wanted to have a look at, okay, what are CD8 T cells doing? So we injected green CD8 T cells, and you can see them crawling through the sinusoids looking for antigen. Because there's no antigen in here, they're not very successful, but they're going to look anyway. And they're quite hardy in how they actually do it. And what if we put a parasite into the system? So you've got the green parasite here sitting in the center. You've got antigen-specific CD8 T cells and non-antigen-specific CD8 T cells. And you can see that as they're crawling around randomly, what they do when they actually find the parasite is they're like, okay, I recognize this antigen, I need to kill you. And so they hang around until that parasite's dead. And that stops, that's the CD8 T cell mechanism of blocking blood stage infection. Now, I mentioned quickly traversal. Now, this is an essential process for the parasite to do. It needs to get into the hepatocyte from the blood before it can actually establish infection. And what there are some bodies of work out there suggest is that it actually gets in, goes through a few before it actually establishes this inclusion body, essentially, to become blood stage. So could we use the techniques that we had to see this actually happening in real time? Yes, we certainly could. And the way that we did it was we injected sporozoites that were fluorescent green tagged. We chucked in the antibodies Cis43 and MAB10, and then we threw in a autofluorescent sugar, essentially, into the system so we could see the vessels. And then we did 2-photon microscopy. And what did we find? I'll quickly pause the video. I want you to keep your eye up on the top left-hand corner. You've got a sporozoite here that's sort of stuck, potentially by a CD8 T cell. You've got a liver cell that's already been busted, so all the dye rushed in. But here you've got a hepatocyte that hasn't been traversed. So it's completely healthy, but then you'll see a sporozoite come through, burst through the system, burst through the membrane, and then all the dye rushes in. So that's traversing, and it's continuing to traverse until it finds the ideal host. And this is really exciting for us. This is a little bit more contrast. Again, you see it happen again. And there's the nucleus, there's the cytoplasm, and the dye has come in through the circulation, and then it tends to just fade away. So yes, we can see traversal. What does this look like if we actually quantify it? So what we found was we used a control antibody, which was against HIV, and you find that there's a lot more traversed hepatocytes than you have with your Cis43, your MAB10. So they're really good at stopping the sporozoites from crossing through. We also characterized whether it was traversing, had established infection, or whether it was still just stuck in the vessel doing nothing. And you find that MAB10 and Cis43 are much more efficient at actually stopping productive infection from occurring, and Cis43 is slightly better at stopping traversal. If we take the dose from 300 to 30, you find that MAB10 loses its ability, but Cis43 still keeps it. So you only need a tiny amount of this specific antibody for it to work. And then again, we used a fluorescent antibody to see how it was binding. And what you can find was something really interesting in that you have the sporozoites are bound with a purple antibody. And what they do is they shed off all their skin. So it leaves a sort of snake skin through the system, and it continues to either traverse or it dies. And Cis43 changed the shape of it, so they ended up sort of closing in on themselves, and they can't do very much at all. This is just a schematic of exactly how we actually did it with different types. And again, you see Cis43 causes this rounding, and you have the parasite shedding its skin, and they both do different things to the sporozoite, but it tends to both actually stop them. And this is not very motile at all. So you can see how it's bound, it's just not doing very much. So what are the immune effector mechanisms of these antibodies? Well, we've decided that Cis43 seems to be an invasion blocker, but MAB10 tends to opsonize the sporozoite and cause it to die. So that leads us to sort of say, okay, there are potential for these two antibodies to be used synergistically. And this is actually going to allow us to design better vaccines to generate these antibodies so that we can actually induce better specificities. And note that RTSS actually lacks the Cis43 epitope, so that might be a good start. And it could also be useful for potential in prophylaxis, especially with people that go to these regions for work, essentially. And finally, I'd just like to acknowledge the funding bodies as well as the really supportive groups that I have and the ANU Medical School. Thank you very much.
So that draws the session to a close. So let's put our hands together for all the speakers and for an excellent audience. Please try and be back by 11:05 for the next session.
this session we have a few talks, and this session is all about microbiome. So it starts with Mukta Das Gupta, and she's going to talk about the genomics analysis of the E. coli in inflammatory bowel diseases.
Good morning, everyone. I would like to thank Sean for giving me the opportunity to present here. Inflammatory bowel disease is a chronic debilitating condition characterized by relapsing and remitting episodes of gastrointestinal inflammation. It is classically divided into two major forms, Crohn's disease and ulcerative colitis. Three major factors contributing to IBD. They are genetic susceptibility, immune dysregulation, and environmental trigger. Plenty of studies implicating bacteria in the pathogenesis of IBD. However, E. coli is found to be the leading candidate bacterial trigger. Several studies have reported a higher prevalence of E. coli in IBD compared to healthy control. Therefore, our aim was to identify the genes and molecular properties of E. coli associated with patients with IBD, adult versus pediatric IBD, lymph node, and aphthous ulcer isolates. This study is based on a total of 179 E. coli isolates from Crohn's disease patients, ulcerative colitis patients, and healthy control. These were taken from different tissues and sources such as intestinal mucosa, aphthous ulcer, which is the early lesion of Crohn's disease, lymph node, those were collected from bowel resected samples. In previous study done by our group, we have found that each human being on an average harbor 3.5 E. coli strains. So to identify the E. coli strains, we have done the RAP-PCR fingerprinting. As you can see in this gel picture, the E. coli produce different banding pattern here. And based on this banding pattern, we have then identified the E. coli strains. We have done the Clement PCR to identify the E. coli phylogroups. Then we conducted whole genome sequencing on a MiSeq platform, did the bioinformatic analysis to identify different molecular properties of E. coli isolates. We have also done the pangenome analysis based on which we identified the gene presence absence among the E. coli isolates and also checked the phenotype-genotype association. We have used different web servers and tools to identify various molecular property, including bacteriocins, capsules, and plasmid, and so on. I have highlighted these molecular properties because I'm going to focus on these properties later in my talk. To avoid the gene bias, we considered only one representative of each unique sequence type per patient. So finally, we have found a total of 146 E. coli strains with unique sequence type. The most common sequence type across all the isolates was ST95, and which is commonly found in human. And this sequence type, no sequence type was not overrepresented significantly in IBD. This is the phylogenetic tree of 146 E. coli isolates. This looks very complex, but I'll try my best to simplify it. The innermost ring here is representing the name of the isolates and the different color indicating different groups. So red for adult CD, orange for pediatric CD, green for healthy control, and blue for ulcerative colitis. Here you can see the healthy control isolates, that's the green isolate, were interspersed throughout the phylogenetic tree, indicating that the IBD isolates were highly heterogeneous. It didn't form any distinct cluster. And the surrounding ring representing different molecular properties, this surrounding ring, such as serotype, phylogroup, plasmid, and antibiotic resistance. Here you can see I didn't find any association of this molecular property with IBD isolates. We found a list of genes significantly associated with Crohn's disease E. coli isolates compared to healthy control. They are plasmid-associated genes and FIMD-associated genes. Plasmid-associated genes are involved in plasmid maintenance, post-segregational killing of plasmid-free cell, conjugal transfer of DNA between the bacterial cell. I'll discuss more about this CCDB gene later in my talk. And the FIMD-associated genes are involved in adhesion of E. coli to the intestinal mucosa of the host. We got very interesting outcome when we checked the association of E. coli, association of different genes in E. coli of lymph node isolates. In addition to a bunch of adhesion-associated genes, we found some gene involved in hydrogen peroxide scavenging and phosphate binding. The hydrogen peroxide scavenging gene encodes an enzyme that has catalase and peroxidase property. The hydrogen peroxide scavenging gene, that is the hydrogen peroxide, is the major reactive oxygen species produced in phagolysosome in response to intercellular bacteria, and which is highly toxic to microbes. So to scavenge hydrogen peroxide, lymph node-associated E. coli likely to use this enzyme that enable them to survive within the phagolysosome. The genes that involve in phosphate uptake also found to be involved in adhesion to the human and mouse macrophage and thereby mediate the phagocytosis. Bacteriocins is the peptidic toxin that produced by bacteria and which inhibit the similar or closely related bacteria. So in the bacteriocin analysis, we found that the botryomycin bacteriocins were significantly associated with pediatric CD and lymph node CD compared to the healthy control. When we checked the association of different capsule type with IBD, we found that the ABC capsule was significantly associated with pediatric CD compared to adult CD and healthy control. Similarly, the group 4E capsule type was also found to be significantly associated with lymph node E. coli isolates compared to healthy control and pediatric CD. As we found some genes involved significantly associated with plasmids, then we used a new approach of plasmid analysis using 3 different tools, and it's a combination of 3 different tools. And the predicted plasmids was then verified again by the MOV alignment against the reference plasmid, and finally confirmed to be present within a strain of E. coli. Here, in the pangenome analysis, we found that the CCDB gene, which is the plasmid-associated gene, that's plasmid maintenance gene, significantly associated with the CD isolates. When we did the plasmid analysis, we found that the PUTI89 plasmid, as well as the CCDB gene that is present within this plasmid, was significantly associated with the pediatric CD isolates compared to the healthy control. So our findings suggest that different molecular properties and different genes of E. coli associated with the pediatric CD, adult CD, as well as the lymph node associated E. coli isolates. This property may allow the E. coli to evade the host immune response in the patients with CD, thereby enabling them to survive within the phagolysosome and maybe helping disease progression. So future study in our lab involves in collecting the macrophage from the blood of CD patients and co-culturing them with these E. coli strains and observing or characterizing the host macrophage E. coli interaction. I'd like to acknowledge the contribution of my supervisors, the collaborators, and Endeavor Postgraduate Scholarship Program. Thank you all.
Beautiful talk and beautiful timing. Thank you very much for that. So because we are running out of time, we have time for one question, if there is any burning question in the audience. If not, we need to move on. Yeah, thank you very much. So the next speaker will be Shasha, and where's Shasha? Yeah, cool. And she's going to talk about the factors involved in pro-operative deaths in colorectal cancer patients.
Good morning. Today, I'm very happy to be here to talk about the application of artificial neural network in cancer outcome research. Artificial neural network is a machine learning approach that works away at human brain to provides the ability of developing new informations by supervised learning and training. It can also be able to deal with complicated prediction model in the big data environment and has been proposed for clinical outcome prediction in many cancer-related settings. So today I'm going to take colorectal cancer as my study focus, as it is one of the leading causes of cancer-related healthcare burden in both world and Australia. We can see from this figure that Australia has high mortality and incidence rate in the world compared to other regions in the world. The darker the color, the higher the mortality and incidence. What we know now, we know that those surgical CRC patients require advanced surgical care, although with some advancement in medical and surgical care, the mortality of CRC patients has been decreasing. However, surgical CRC patients still experience elevated risk of post-surgical mortality. And these adverse outcomes are highly related to a range of clinical and social demographic factors. It is commonly known that some fatal comorbid factors such as cardiovascular disease and their social demographic factor like age are related to their poor post-surgical outcomes. But what we don't know that which one is relatively more important. Like coexisting cardiovascular disorders versus mental disorders, their private insurance versus their age. So my research question is, across those factors, which one is relatively more important, as those findings might inform countermeasures to improve their post-surgical outcomes. We use the census of inpatient data records in New South Wales and focus on surgical CRC patients during their acute care. These are the factors of interest that we used in this study. We also considered a range of comorbid factors that including infection, blood immune disorder, endocrine, mental, and so forth. We developed 3 layers perceptron backpropagation neural network in this study that include one input layer, one hidden layer, and an output layer. Compared to conventional logistic regressions, it has advantage in many complicated scenarios such as nonlinear relationships, high-ordered interactions, and interrelated causal pathways. So it also can have some better predictive performance. From this matrix, we can see that the factors of interest in our study are highly correlated. So logistic regression would fail the model specification, as we did a specification link test to see whether logistic regression can pass the test, but it failed. But AN works well in this setting. We developed an AN schematic scheme. This is the one we used in our study. We can see the high-ordered correlation between those predictive variables. We also ran a 10-fold cross-validation to identify the optimal neurons in this model. It's 14 that's hidden layer to prevent overfitting, because there's a common condition in the AN model is it might have overfitting conditions, but if you did 10-fold cross-validation to prevent that would be fine, and also to improve their accuracy of prediction. We separate the patients in our study into 2 groups based on their Charlson Comorbid Index, as we want to see how the relative importance would vary across the different groups of severity, comorbidity severity. So the one group with less severe comorbidity, the other with severe comorbidities. For patients with less severe comorbidities, it seems that the musculoskeletal disorder is relatively more important than other factors. Whereas for patients with severe comorbidities, it is still related to mood death, but relatively less important than a couple of other factors, such as infection, the rural residents. This is the full table for the relative importance of all factors of interest. Those protective factors are marked with asterisks. We can see here the relative contribution of each factors that change across different groups of complex comorbidities. So we can see that musculoskeletal disease is one of the most important predictors in this model. Well, I think the importance of some conditions like their complication, their infection, post-surgical outcome are understandable. But for musculoskeletal disorder, I think I still need to speak a little bit more about this. Well, in our study, most of the CRC patients, they had bone mineral density disorders, which is the predictor for post-operative mortality, and one of the biomarkers for CRC progression. So we can imply that this might indicate that those patients with comorbid musculoskeletal disorder may have advanced stage of CRC. So they might require complicated surgical care. Previous study also identified the benefit of using drugs for treating osteoporosis can help to reduce post-operative mortality for CRC patients. So that will be helpful if when you focus on those people with comorbid musculoskeletal disease, you prescribe them some medication for osteoporosis. On the other hand, they can also help to improve their post-surgical outcomes after CRC surgery. Another factor I want to talk about is mental disorder, as it's become the fourth most important factors in patients with severe comorbidities. Cancer patients with mental disorder experience elevated risk. Someone may develop the mental disorder before their development of their cancer or develop mental disorder during their cancer treatment. So this might imply that the provision of psychosocial intervention or some medical support may be necessary for improving their outcomes. Like when there's some drug-drug interaction between mental disorder treatment and the cancer treatment. So if you, when you prescribe them some medications to treat mental disorder or cancer, you must be reconsidered as it may aggravate patient's illness. Well, this study provided insight into the relative importance of coexisting conditions in relation to post-operative in-hospital death and how they vary across different severity groups. This may assisting in assessing risk of surgical intervention and providing appropriate treatment procedure or strategies to help for people with complex needs. Also can help to improve patient-centered perioperative care for CRC patients, targeting specific conditions like musculoskeletal disease or some social demographic disadvantage might help to improve their perioperative care. We have some limitation in the study. We only considered in-hospital death, that's the short-term outcomes versus long-term, like 30 days the post-surgical care. In future, we need to examine other adverse outcomes identified in our study and applying in other settings for prediction and develop future prevention strategy to help with those people with complex needs. And last, I would like to express my special thanks of gratitude to my supervisor and other co-authors for their support and contribution, and appreciate New South Wales Department of Health for providing the data. Thank you very much.
Thank you very much for keeping on time. If there is any burning question from the audience, one question we can take. Rushing through it. No? I'm just curious about that because you were talking about the socio-demographic factors. Would you consider like the economic status of the patients will be have an influence, like because you took into account like a mental factor or being single or not.
Yeah, we can
You
see
took the rural area into account as well.
Demographics data, they are a kind of protective factor, which means with a higher stage of their SES, patient will less likely to experience poor post-surgical outcomes. And many studies identify that their outcomes are highly related to their SES. This is important, but compared to other factors in this, it's less likely to be just more significant.
Yeah, excellent.
Thank you.
Well done. Let's thank Shasha again. So I would like to invite Claudia Slimings. Claudia? Claudia is going to talk about the social and community influences on dietary attitudes.
Thank you. Good morning. Thank you for inviting me to speak today. Can you all hear me okay? Thank you. So my name's Claudia Slimings. I'm from the ANU Rural Clinical School. I'm actually presenting today on behalf of my colleagues, particularly Marie Smith, who initiated this work with her supervisor, Sarah, as part of her research project requirements for her medical degree. And she's currently on a rural placement, so she can't be here today to present. So in Australia, we're quite lucky. We actually experience a relatively high level of health. But like most high-income countries globally, 1 in 2 Australians have at least one of the 8 most common non-communicable diseases. We know that cardiovascular disease is the leading cause of death in Australia, and that obesity is one of the key risk factors for cardiovascular disease. And the most recent data, 67% of adult Australians are overweight or obese. We also know that burden of disease varies by certain population groups. And in rural communities, the prevalence of many non-communicable diseases is higher. So for example, there's high prevalence of cancer, diabetes, coronary heart disease, and also overweight or obesity. So nutrition is a key behavioural determinant of obesity and chronic disease. And access to healthy food is influenced by a number of individual and area level factors. And these may be amplified by living in a rural community. So for example, population size and remoteness may combine to limit people's access to healthy foods in terms of the cost of food or the health of the food outlets available in the communities. There's also a range of individual level factors. So of course, education, occupation and income feature. But one that we're particularly interested in is social capital. Now, social capital is the social networks that people have within in society that may influence their behaviour or opportunities. And it's particularly of interest to us in working in rural health because rural communities tend to have quite close-knit social networks. So the aim of this study was to determine associations between social and economic characteristics, dietary attitudes and fruit and vegetable intake for people living rurally in New South Wales. This was a community-based cross-sectional survey that ran between February and July of 2018. Participants were predominantly recruited from rural shows that took place in 4 towns in southeastern New South Wales and supplemented with data from 2 general practices and 2 other towns. And participants completed a paper-based questionnaire. This slide just summarises the types of topics that we covered. So we looked at diet, economic factors, social capital, and of course some demographic factors. In terms of diet, we asked respondents to report how many cups per day of fruits and vegetables they ate. And this was then approximated to align with the NHMRC Australian guidelines on fruit and vegetable consumption. We asked a set of 5 statements about attitudes towards diet, and the responses to these were summed with a range between 5 and 25, so having a higher dietary attitude score meant that they had healthier attitudes towards nutrition. For social capital, there were a couple of questions that we asked. The first one was based on work that was undertaken in the Social Science Department at ANU, looking at social class in Australia. And one of the social capital indicators they came up with was presenting people with a range of 21 occupations of varying prestige. And participants had to select who they knew from those occupations in their social networks. So not the accountant that they employed to do their tax return, but who they actually knew socially. And so the sum of those known occupations indicated you had a more diverse set of people in your social networks. We also asked about people's spaces that they socialised in. So we asked how often they socialised in cities, big towns or small towns, with the latter meaning if you were more likely to socialise in your small town or your local community, you were more engaged socially with that community, as you had greater social capital. In terms of data analysis, we used linear and logistic regression models to analyse three main outcomes. So we looked at adequate fruit consumption, adequate vegetable consumption, and also the dietary attitude score. Now, in the models of fruit and vegetable consumption, we also included the attitude score as a predictor. So this just summarises the demographic and social results. So we had 326 respondents. Median age was 57 years, 64% were female. 89% lived in inner regional areas as opposed to outer regional areas, and nobody was in a remote area. A large proportion, 41%, were economically inactive, and 69% of these were actually retired. And that probably reflects that the people who came to the rural shows were likely to be sort of older people who engaged with who enjoyed going to shows. So this summarises just the dietary outcomes. So 47% and 39% of respondents met the fruit and vegetable guidelines respectively, and the median diet score was 19, ranging from a minimum of 9 to 25. So these are the logistic regression results for fruit and vegetable consumption. So in terms of fruit consumption, the odds of eating or meeting fruit consumption guidelines increased by 13% for each unit increase in the dietary attitude score, and the meeting vegetable consumption increased by about 19% for each unit increase in dietary attitude score. Now, the socioeconomic factors weren't so strongly associated in these models because they were probably overtaken by the more proximal risk factor of dietary attitude score. In the unadjusted models, a number of the socioeconomic factors were related, but once in the multivariate models, their influence was weakened. This is the linear regression results for dietary attitude score. So we found that the occupations that you knew socially, as well as socialising in small towns, was associated with a more positive dietary attitude score. The associations were quite modest, so the score increased by 0.07 points for each additional occupation that was known socially. So this means basically somebody who knew about 10 occupations, their score was somewhere around, if I can read off the graph, just under 19, but if you had more than double, or if you had double the number of occupations known socially, say about 20, it was about 19.6, so a very, very gentle relationship. We also found that the attitude score increased by 0.9 points, or nearly a point, for people who regularly socialised in small towns compared to people who didn't. What was interesting was that we found some gender differences, but the power of the study wasn't really quite large to make firm conclusions. But what it indicated was that the occupation relationship, or your networks of occupations, was stronger for men than for women, but the reverse was true for women. So those who socialised in small towns, there was a stronger relationship for women than for men. So we concluded that greater fruit and vegetable intake was predicted by healthier dietary attitudes, that in turn the healthier dietary attitudes were related to more diverse social and community connections. And so this aligns with current literature that social network comprised of individuals from a diverse range of occupations is likely to expose individuals to a variety of perspectives, and that socialising in communities with positive dietary attitudes may spread positive dietary norms. And so these provide new insights on the importance of social and community networks in the Australian rural population on dietary attitudes, and may offer some opportunities for health promotion activities through engaging through local community networks. Of course, our study has limitations, so this was a volunteer sample, and the study wasn't representative of all South Coast New South Wales or South East New South Wales residents, so we had an overrepresentation from inner regional areas, an older sample than people living in rural areas in the whole of New South Wales. And of course, there's always some errors in your measurement, so people having to remember how many cups of fruit and veg they ate on average on a day, and trying to approximate that to the Australian guidelines was a bit of a challenge. But I'll finish on that note, so thank you very much for listening.
Thank you, Claudia. Nice study. Any questions? Hannah?
Is there any reason why you had a lot more women at the age responding to your survey? Are we following a trend where men just don't do surveys, or is it just there's more of them alive
We can
and ready to
hypothesise,
answer?
but who knows? Who knows? I think women are generally more likely to participate in surveys. So
they skew all our results.
Yeah,
within,
so
don't they?
the rules shows what happens is the Australian Royal Medical Students set up a blood pressure testing tent, and people can come up and be tested. And so as part of that, our students running the surveys will try and recruit people to fill in and form theirs. It may be that women tend to engage with health services more anyway. So, yeah.
I have one question. Did you take into account that what type of vegetable, what type of fruit they consume more?
No, it was a very, very superficial question. We are doing another study where we're asking a bit more information about people's eating habits here.
Excellent. So let's thank Claudia again. So the next speaker will be Louise Brightman, and she's going to talk about the patient's factor in a publicly funded obesity program.
Good morning. Actually, I'm not actually Louise Brightman. Dr. Brightman is out there in Alice Springs practicing psychiatry. So I got the job of actually presenting this paper for her. So today we wanted to talk about some of the outcomes and some of the factors that determine our patient attendance and access to interventions in the Canberra Obesity Management Service. So I think it's no surprise to everyone that obesity is the leading cause of chronic disease globally, and that the prevalence of obesity and its associated health burdens have continued to rise over the years. And several Australian states and territory health departments, including the Canberra Health Services, have introduced a publicly funded specialist obesity service. And so the Canberra Obesity Management Service was initially launched in 2014. It is a comprehensive management program for adults with class 3 obesity with a presence of a comorbidity. And the aims of the service is actually to improve overall health outcome rather than having a weight-centric sort of focus. We accept referrals from medical practitioners, and it is led by a physician, but it is multidisciplinary in nature, and we use a chronic disease management approach with individualized case management. And our patients construct and implement an obesity management plan with the support of medical, nursing, and allied health staff. And we have the capacity to facilitate intensive interventions for selected patients with appropriate medical supervision. So the aims of this study, typical, is to determine the baseline characteristics and the pre-existing comorbidities of our patients, try to assess the degree of multidisciplinary engagement and access to allied health services and intensive interventions. We also wanted to have a look and see if any of the measurements changed over time and to determine what proportion of our patients achieved the clinically significant weight loss. We looked at their depression and anxiety scores as well as their self-reported sleepiness scale, and we also wanted to have a look at the prevalence of sleep disorder breathing in our population. I'm sorry, switchboard continues to ring me. So the methods we have used, this is a descriptive observational study using retrospective medical records review. And the inclusion criteria is all new patients who presented to the service over a 12-month period. So this is essentially a consecutive series of patients that walked throughout doors. And we excluded those who have already been seen prior to this period, and all those who have been referred outside this date range. So over a 12-month period, we welcomed 162 patients to our service where they met the eligibility criteria and attended an initial review with a physician. And as you can see here, the mean age of our patients are around 45, with 73% being female. The mean body mass index in our population is 49.5, and the mean weight is 139. And in addition to their class 3 obesity, the average patient had another 3 comorbidities. And we then stratified those into those who continue to engage beyond seeing the doctor for the first time and those who did not. And those patients who continue to engage within the service have a slightly higher body mass index and perhaps has more comorbidities. So most of our patients, apart from their obesity, has a significant number of comorbidities, and I think this is no different from most of the obesity services around the world. Hypertension, depression, and sleep apnea and diabetes are all very common in our population. So what did I do in our service? 103 out of the 162 engaged beyond the initial physician assessment. So about 36% of the patient decided not to engage any further beyond seeing the doctor for the first time. And wherever possible, we asked them why they decided not to continue. And a few were actually, in fact, they're not in class 3 obesity. Their body mass index were less. And there were a few who are actually obviously lacking capacity to self-manage. But often we have patients who request discharge because for a variety of reasons, including unable to implement behavioral changes. They're seeking surgery and isn't really interested in anything else. Some were not aware of the referrals or have transport issues. Many had cite other competing priorities, and some of them were just simply unable to find them. And self-reported sleepiness or symptoms of depression, anxiety, or stress did not actually predict attrition. So of all the patients, 8% declined to see any of the allied health staff, but most of them utilized at least one service, and 22% engaged with all 3 services, which is dietetics, psychology, and exercise physiology. And 37% of the patients also, in conjunction with their physician, underwent intensive interventions, which could include very low energy diet, pharmacotherapy, and private bariatric surgery. At that time, the publicly funded bariatric surgery is not really available in the ACT, but this has changed since that time. So what happened to our patients? The mean change in weight was actually 5%, and 36% of our patients achieved the clinically significant weight loss, and they are shown as the purple bars on the left. And with about 19% losing more than 10% of their body weight. And a further 47% demonstrated weight stabilization, and they are the green bars, and a small handful in the red on the far right of the graph are the ones who continue to gain weight. And if we actually look at the graph on the right side, those who had attended initially and then did not actually engage subsequently seems to have a lower BMI, but those who have a high BMI seems to engage for longer and probably have a greater reduction in their body mass index. Now, as I'm a practicing sleep physician, I have an interest in sleep disorder breathing. So what we have implemented is in our service is we embedded sleep symptoms as part of the initial physician assessment. And we found that 26.5% of our patient comes to the service with a pre-existing diagnosis of obstructive sleep apnea. Another 37% were found to have a sleep related symptoms and were referred for a sleep study. And of the 37% where we feel has significant sleep symptoms, 87% turned up with a new obstructive sleep apnea diagnosis. And interestingly, of these patients, 17% actually had obesity hypoventilation syndrome, which is defined as having awake type 2 respiratory failure with hypocapnia on an arterial blood gas. And in fact, if you look at this, this actually makes obstructive sleep apnea our #1 comorbidity. And it is clear that perhaps we have much more work to do in the primary care space because more than half of the patients with sleep disorder breathing were undiagnosed prior to coming to the service. So in conclusion, 36% of our patients achieve a clinically significant weight loss, and most of our patients have engaged with multiple allied health services, with 37% also using an intensive intervention. And those who have a higher body mass index engaged for longer and probably have greater body mass index reduction. There are multiple psychosocial barriers that prevent patients from engaging with our service, and obstructive sleep apnea is the most common comorbidity, and they remain undiagnosed prior to presentation to the Canberra Obesity Service. And we like to think we present this report to support a chronic disease management approach to obesity, and hope that this may serve as an example of data collection, individual service monitoring, and also future comparison across multiple specialist obesity services. And finally, I'd like to acknowledge my co-author, Dr. Louise Brightman, who did a majority of the work, and Dr. Paul Dugdale. And it's great to work in a team, and here's our team within the Obesity Management Service. Thank you.
Thank you very much. We don't have time for any question. If you have any question, please catch her after the session. We need to move on. Thank you very much. So the last speaker for the individual research talks is Natalia Martin. She's going to talk about the inflammasome activation by E. coli and relation to inflammatory bowel diseases.
Good morning, everyone. So as Fazi just said, I work in the area of inflammatory bowel diseases, and today I'm going to talk about an ongoing study looking at how the host immune system responds to bacteria or E. coli isolated from patients with and without IBD. So IBD, or the inflammatory bowel diseases, result in relapsing chronic inflammation in patients, and it's largely incurable. There are 3 core contributing factors currently under investigation, and my research focuses on 2 of these. The first is the intestinal microbiome. So although no single bacterium is known to be causative, E. coli is a leading candidate and is the bacteria of interest in my studies. I'm particularly interested in how these E. coli interact with the immune system, particularly looking at characterizing the inflammatory response pathways. So the inflammasome pathways play a critical role in our immune system. They form these multi-protein inflammasome complexes that play a key role in cell inflammation and cell death in response to pathogens. Importantly, dysregulated inflammasome activation has been linked with intestinal inflammation in animal models. So the nod-like receptor protein 3, or NLRP3 inflammasome pathway, is the best characterized and responds to a diverse range of pathogens, so we chose to start here for our studies. Just to briefly go through how this pathway works, the first step is a priming signal. This is where the cell is exposed to bacterial ligands, such as DNA or LPS, which results in upregulation of NLRP3, but it remains in an inactive form. This is followed by activating signals. So the cell is exposed to pathogen-associated molecular patterns or damage-associated molecular patterns. This results in physiological changes in the cell, such as iron flux, which results in activation of NLRP3 and formation of the complex. This activates caspase-1, results in the activated form of this caspase, which is a cleaved form. The cleaved activated caspase-1 then has two key roles. The first is it results in a form of cell death known as pyroptosis. And secondly, it results in the cleavage of pro-interleukins such as IL-1 beta and IL-18 that are then secreted from the cell and feed back into the system and further activate the inflammasome. So the aim of my work was to characterise the NLRP3-dependent inflammasome activation by E. coli isolates from IBD and non-IBD patients. Just to briefly go through the methods that I used to achieve this. So the first step is to take bone marrow cells from both wild-type and NLRP3 knockout mice. These bone marrow cells were then differentiated into mature macrophages using colony-stimulating factor 1 or CSF1. And this is the same factor that is responsible for the differentiation of intestinal macrophages in our guts. These macrophages were then co-cultured with patient E. coli isolates. I then collected the supernatant and protein and looked for NLRP3-dependent caspase-1 activation using Western blot analyzer techniques. So here you can see I looked at a total of 36 E. coli strains from patients. So on the left in red, these 13 strains were from patients that did not have IBD, and the remaining 23 in blue on the right were from patients diagnosed to have inflammatory bowel disease. 20 of these had Crohn's disease, and the remaining 3 had ulcerative colitis. I'd just like to emphasise here that just because there are 36 strains, that doesn't mean they were from 36 patients. The ones highlighted here in shades of yellow are different strains but isolated from the same patient, just different sites of the bowel. So I wanted to identify NLRP3-dependent caspase-1 activation, and this is some Western blot analysis representative of that. So as I've said, I isolated macrophages from both wild-type and knockout mice, and I either cultured these without the presence of any bacteria as a control here, shown as a media control, or in the presence of each of the 36 strains of E. coli that I just went through, and I have 3 representatives here. So the Western blot looked for, firstly, in this upper box here, a pro-caspase band. This is the inactive form of caspase-1, and as you would expect, you can see it across all of our samples. The lower band is specifically looking at the activated form of caspase-1, which is the P20 band, and you can see an NLRP3-dependent activation in 2 out of these 3 strains. So I looked for caspase-1 activation across all 36 strains, either directly using Western blot analysis or indirectly by looking for IL-1 beta or IL-18 secretion using ELISA. So for the 23 strains that were from IBD patients, 18 of these showed both direct and indirect activation of caspase-1. For the non-IBD strains, 8 out of the 13 showed caspase-1 activation by Western blot and 7 out of the 13 by ELISA. I'd like to make a point here that 4 out of these 8 strains had a pathogenic phenotype, that is that the patient had sepsis, a bladder infection, or the E. coli were characterised to have what's known as an adherent invasive E. coli phenotype, which is a pathogenic phenotype. So our lab also complemented this work with some bacterial genome analyses that was performed by a PhD student in the lab. So we have the whole genome sequences for 31 out of the 36 E. coli strains. 23 of these 31 strains showed a caspase-1 activation phenotype. And interestingly, we found that there were 2 bacterial genes that associated with the caspase-1 activation phenotype, 10 strains for the first and 18 strains for the second. And these genes played roles in bacterial toxin production and bacterial protein translation pathways. So in conclusion, E. coli strains isolated from the intestine of patients activated the NLRP3 inflammasome, and we are currently expanding this study to include more non-IBD strains from patients that don't have other pathogenic phenotypes.
Although we didn't see a significant association with the IBD strains themselves, it was interesting to find that 2 of the E. coli genes appeared to associate with NLRP3-dependent caspase-1 activation, which could have implications for other forms of inflammatory conditions. And in future, we hope that characterization of the host microbe response in IBD patients could better inform clinical treatment. I'd just like to thank my lab, our collaborators, and CHARM for giving me the opportunity to speak today. Thanks.
Nice work. Is there any question? So this association has been found before, or that's a novel thing that you found?
Novel.
It's a novel.
Yep, specifically with the inflammasome. Yep.
Yeah, and you're working on the 2 genes that they are associated yourself, or the other members in the group are working on that?
Yeah, it would be the other members in the group. Yep.
Very nice. No more questions? Excellent. Let's thank Natalie. So the individual research session will finish here, and let's put our hands together for all the speakers during the CHARM, and especially for this session. I'm going to hand it over to Claire, and we have the keynote speaker session, Professor Mark Morrison, and the keynote session will start now. Thank you.
We've just got a few minutes to get set up. We'll start the keynote presentation at midday. In the meantime, for those who are coming to the dinner tonight, please make sure that you arrive on time. Maybe get an Uber on the way home.
I've got a hot mic on. So, nothing. Plead the Fifth Amendment already.
And then slide advances, I'm sure you've used this before, that laser pointer.
Yep, there it is.
And then forward, back.
Okay, thank you. I think so.
Okay, so welcome everyone to this keynote talk from Professor Mark Morrison. We've saved the best till last. So Professor Mark Morrison comes to us from the University of Queensland, where he's been since 2013. He's located at the Translational Research Institute campus and he's a chair and principal group leader in microbial biology and metagenomics for the UQ Diamantina Institute. In addition to this role, he now serves as the microbiome science leader for the Translational Research Institute, as well as science lead for gastrointestinal function with the Princess Alexandra Hospital's Department of Gastroenterology and Hepatology. During his academic tenure in the USA, Professor Morrison led the team that produced the first genome sequences for the bacteria Ruminococcus and Prevotella, with both genera now widely acknowledged to play a key role in human gut enterotypes. His ability to translate genomic and metagenomic data sets into a sound biological framework include a world first, the metagenome-directed isolation of a new bacterium from a species-rich microbial community. He is science representative to the International Human Microbiome Consortium and the Chinese Microbiome Initiative, and serves on a number of institute and industry advisory boards for microbiome research. So in other words, Professor Mark Morrison is the microbiome guru, and we're very lucky to have him here today to talk to us. Over to you, Mark.
Thank you. Thank you, Claire and Hannah and the organising committee for the chance to be here. I guess being called a guru is a reflection of age more than anything else. So what I'll try and do today is talk about how the gut microbiome is being examined currently using technologies that really have been advances linked to the 21st century, which involve next generation sequencing technologies. But I go back longer than that, and I was actually trained as a microbiologist, and I think that's my first disclosure. I have PhD training in microbiology. I don't have any medical qualifications, but have benefited greatly from the collaborations I've developed over the last 5 or 6 years, especially since I moved to UQ with clinician scientists and clinical gastroenterologists in particular. And I think what the gut microbiome really is it's something more than just DNA sequence data. It's really looking at the microbial community in a holistic and hopefully mechanistic way so that you can start to better understand the interactions and processes that take place not just within that community itself, but in response to or in communication with the host and the physicochemical conditions that are provided at those different sites on the human body. So if we think of phenotype as the expression of genotype by environment, by lifestyle interactions, I think of the microbiota as the X factor that links those three things together. It's a functional and dynamic interface between those three components that influence host phenotype. But I think now it's much more objectively measured and quantifiable in terms of its capacity and its functionality. And so I have a talk for you today that I think has 4 parts. The first part is an overview in terms of how these technologies have transformed the study of the gut microbiome over the last 10 or 15 years. And then I'll give you a couple of working examples of some of the basic science that my team is leading in collaboration with colleagues in the Australian Gastrointestinal Research Alliance as it relates to upper GI conditions and functional gastrointestinal disorders. And then a second presentation in relation to work I'm doing with Professor Michael Kam and Xu Ning at University of Melbourne and Hong Kong University respectively in relation to examining the gut microbiota of Chinese nationals here in Australia, in Hong Kong and mainland China that develop Crohn's disease, and some of the techniques and approaches we're using to try and learn more about that. And then I'll finish off with what is often the most interesting or widespread curiosity for persons in this space, and it's the impact of diet on the gut microbiome. And I'll try and give you a couple of examples of how you can make friends with salad as part of this overall project. So you've had long enough to look at my disclosures. I'll get on with the talk. So the reality is we live in a microbial world. We're surrounded by microorganisms that provide a great variety of goods and services to the ecosystem and to our natural and managed environments. And the human body really serves as a scaffold, an architecture that has recruited over evolutionary time collections of specific microorganisms that colonize and get a benefit by being associated with that body site And over time, they've developed a relationship which is somewhat homeostatic insofar as it's a degree of detente. They're competing for certain things, but there are also benefits to each partner for their presence being there. And I've shown here on this figure some pie charts, and the colour coding gives you an idea of the proportions of microorganisms that are present. But principally, if in nature these categories of bacterial classification now approach more than 60, only about 6 of those are consistently and commonly isolated or identified associated with the human body. So in other words, it's a rather subtracted and specialised collection of microorganisms that has made the human body its home. And it's not necessarily anything that comes out of what's present in nature. And the relative abundances of these core 6 phylogenetic groups at different body sites are shown in these pie charts here. The surface area of the skin is about 2 metres squared, about the size of a large tablecloth. And per square centimetre, we might have as many as 4 million bacteria. But the large intestine really is the place where there is a residence for a very dense population of microbes, up to 10 to the 10 or 100 billion microbes per teaspoon of luminal contents or stool material. So the density and the quantity of microorganisms that resides within this body site, and given the fact that the intestinal tract itself in terms of the surface area is incredibly large in relative terms and chock full of a variety of different cell types, both structural and immune-based, the interactions that take place between these communities are really quite intriguing, diverse, and to a large extent still cryptic. There may be as many microbial cells as there are human cells in the body. You may hear different variations of this ratio, principally trying to brag how important the microbiota actually are. The reality that I want to bring home to you is that we do live with a microbial community that shares this environment with us, and to a certain extent our genetics have an influence over that. So too does the environment that we live in, and also the lifestyle choices we make, whether we consume alcohol, whether we smoke or not, whether we do exercise. All of these features can have influences in terms of this relationship that develops between our own cells and the microbial world. So where am I going with this? This is the thesis published by Theodore Escherich when he was talking about pediatric gastroenterology and the bacterial basis of infant diarrhea. And it was around about the time when Koch's postulates were really starting to develop. And he kind of questioned why he was working in this space, where you had the development of Koch's postulates and someone working on stool samples and looking at the microorganisms in there. There were thousands of different types, a situation that seems to be controlled more by a whole range of coincidences rather than mechanistic interactions that may occur. But even back in the late 1800s, he felt that not only were this group of microorganisms important to food digestion, but they do offer some involvement at least, not necessarily always the cause, but some involvement, some control over the volume switch associated with some of the chronic non-communicable diseases that we see in the world. And as lifestyle and environmental changes have taken place over the course of the 20th and 21st centuries, the increasing incidence of these non-communicable diseases within the framework of Western populations in particular, where diets, antibiotic use, and the hygiene hypothesis have all become quite widespread, broad cultural practices reflective of these sorts of populations and communities. So what was the challenge? Well, when I was a PhD student, our understanding of the microbial world was based on pure cultures. It was what you could grow in the lab that you studied, and of course Escherichia coli was in relative terms, technically quite straightforward to grow. And a lot of work was done with this organism in terms of developing techniques in genetics and molecular biology approaches that have been really instrumental in driving the field forward in the general area of cell biology. But we also knew, even if you just looked at the samples under the microscope, that the majority of microbes in nature were resistant to culture. I'm not going to say that they're not culturable. I'm just going to say that at the time, with the technologies available, the capacity to culture these organisms axenically by themselves was extremely difficult to achieve. But the next generation sequencing technologies, I think, have opened up some windows in terms of looking at these complex and diverse mixtures, but also looking at them within the framework of the actual place where they originate from. So the opportunity now to look at tissue samples collected during biopsies, or skin swabs, or blood samples, these techniques that are collectively referred to under the umbrella of metagenomics provides us an opportunity to look at this microbial community in a much more holistic and mechanistic way. And through DNA sequencing and basing your work initially on DNA extraction, the step required around cultivation is precluded. You don't need to culture anything to get started. You just need to be sure that you capture the microbial community in a non-biased and holistic way and undertake an appropriate means to extract the DNA or RNA from that sample, and then use DNA sequencing technologies like we do in human genetics or genomic medicine to provide insight in terms of structure and functional variations associated with that composite set of genetic information. So how do you do it? So here's your microbial community sample here, and as we go further into the talk, you'll see that I'm a microbiologist that's learned how to use these techniques and not the other way around, okay? So I'm one of the throwbacks where I come from a background in microbiology, and I've learned how to take advantage of these techniques as part of my overall research approach. For a large number of people working in the community of metagenomics or microbial genomics, it's the other way around. They've learned their science based on DNA and RNA sequencing, and now they're showing an interest in terms of how to get involved with microbiology. So in this talk, as I go through some of the basic science, I hope you see the value and opportunity that's involved with being able to isolate microorganisms and study them. A couple of the presentations, especially from Claire's group this morning, is a good working example of that, but I'll give you some more with a greater reach out in terms of the biodiversity associated with these samples. And so there's really two methods. One's called 16S-based profiling, and the other is called shotgun metagenomics. And this technique here, going north, is the most widely used and still the most widely published. And what's involved here is that the community, again, you don't culture the microorganisms necessarily, you just need to ensure you capture the community in a non-biased and holistic way. You extract DNA from it, and then by PCR you amplify the region of a universally conserved gene that encodes for one of the ribosomal RNAs. And that region, then that amplicon can be sequenced, and because there's a dictionary out there that has an incredibly large number of these sequences relating to specific bacterial taxa, you can start to interpret what types and kinds of microorganisms are present in your sample. And then by using various types of mathematics and statistics, start to get a compositional assessment of what types and kinds of microorganisms you have present in the individual samples. And so if you've done, or your samples have come from a case control study or some sort of cross-sectional assessment of persons with active disease compared to healthy subjects, it gives you the opportunity to compare those communities with each other to see how similar or how different they are. And so mathematically, you end up with data sets that look like these two here. These are generated from projects that I've been involved with, looking at patients with active Crohn's disease compared to healthy controls. And you can see that one group, the healthy group, is here in green, and the IBD or the Crohn's disease group is here distinctly separated, suggesting that the community profile and the variation within it separates or clusters these persons according to their health status. And then when we look at each individual bacterial taxa, as shown by these coloured bars over here, and the relative numbers of them, we start to be able to produce a heat map for each individual person, demonstrated by the columns here, and each row represents a different type of microorganism, in this case bacteria. And you can see that for the healthy cohort, principally there is a collection of bacteria in relatively high abundance, which are in relatively low abundance in the Crohn's disease group, and vice versa. There is a collection of bacterial taxa that are in high abundance in patients suffering from Crohn's disease, and in healthy human subjects, they're in relatively low abundance, suggesting that there are changes in the community profile in response or associated with the physicochemical conditions associated with disease. And you can start to differentiate your patients and potentially come up with biomarkers that are indicative of disease risk or disease status. I'll use these examples here to talk about shotgun metagenomic sequencing. And so in this case, we take the DNA, break it up into small pieces, and you construct it in a library that then is compatible with the Illumina sequencing technologies of the day in particular. And just like you would with exome sequencing or whole genotyping for human DNA samples, you will produce large quantities of data based on those libraries and then separate those reads into the different sample groups, and it provides you with a functional assessment. It's non-biased, there's no culture-based step, but in this case, rather than a PCR step just focusing on one single gene, you now have gigabase pairs worth of data representing the functional characteristics of that entire community. And so it's giving you an insight in both what types and kinds of microbes you have, but also what changes in the functional capacity or potential exists within those communities. And I use these three papers as good examples of this. This is collaborative work done by groups in Europe, particularly the French group at INRA, with Beijing Genomics Institute in China. And basically what they have is 150 patients are considered to be healthy controls, and about 150 patients being diagnosed with type 2 diabetes. A stool sample is being collected from each individual, and shotgun metagenomic data is being produced from each sample, representing 150 data sets here on the left to compare to 150 data sets here on the right. And so by using various bioinformatic tools, you can start to build a picture in terms of how similar or how different the microbial communities are to each other. And the size of the circle and the colour of the circle are indications of the population size. The bigger the circle, the greater the relative abundance. And likewise, the colour of the circle gives you an indication of what type of microorganism it is. And you can see that the coloration of the circles as well as their sizes are really quite different between the control subjects and those that suffer from type 2 diabetes. And there's a range of positive correlations shown here in blue and negative correlations shown here in red that help to differentiate these communities. And then you can take the rest of the data, and shown here in blue are the functional attributes of the community that appear to be enriched in the healthy subjects. And here in red are the functional characteristics of the community that appear to be enriched in those patients suffering from type 2 diabetes. And as a result, you can start to see that there are variations in the metabolic potential of the communities between the two types of patient or cohort groups. This was subsequently identified as influencing an economy associated with the production of branched-chain amino acids and their breakdown into branched-chain fatty acids, and influencing that economy influenced the amount of branched-chain amino acids reaching serum, which was subsequently shown to have a modulating effect upon the efficacy of insulin for glucose clearance. And so there was an interaction identified because of these types of functional data, looking at the branched-chain amino acid economy in type 2 diabetic patients. And then my colleagues in China, Liping Zhao and Chen-Hong Zheng, published a recent paper in Science that showed by an intervention of Chinese traditional medicine and the provision of fermentable fibres, you were able to recalibrate this community more towards one that looked like this, with these sorts of attributes, and alleviate at least some of the symptoms associated with type 2 diabetes. So one of the first kind of demonstrations of how diet could be used to manipulate the gut microbiota with positive outcomes in terms of some of these metabolic parameters. So shotgun metagenomic sequencing provides new opportunities and great insights in terms of structure and function of the community. And its application with stool samples or samples where the density of bacteria are very high is extremely effective, okay? Where constraints or limitations still reside is those sub-communities that are associated with tissue, or their diversity is low, or their density is small. And as a result, human DNA is the principal component of the total DNA that you recover. And while you can use PCR to get around that, to get some picture of the microbial taxa or profile, you're limited by how much sequence data is produced from the human sources of DNA rather than the microbial population. So I'll deal with that in a moment. One of the last points I want to make before I move on from here are these white circles here. And you can see that there are a variety of white circles, not just in the patients with type 2 diabetes, but also in the healthy control group. And what these white circles represent are organisms that even still remain unclassified. In other words, they're representatives of bacteria that may occur within the stool microbiota of healthy humans as well as those with type 2 diabetes, but we've not yet isolated a representative of those microorganisms to understand its metabolic features or its capabilities. And so this is where microbiology in a traditional way has something to offer in this space, and I hope I'll give you a couple of examples where that's been working in our group. So the first presentation or short story I want to tell you about is work that I do in collaboration with the Australian Gastrointestinal Research Alliance, and it's to look principally at functional GI disorders. And the driving force for this is that currently the approach is really to try and manage symptoms in functional GI disorder patients, and often it's not done very well or for very long, and undergoes a rather long and lengthy process that really doesn't help people deal with their symptoms. And so often, maybe as many as 1 in 3 persons report symptoms, 1 in 6 go on to have specialist consultations, and the majority of them are negative endoscopically. And so the opportunity really is around the use of a variety of different pharmaceutical-based interventions to try and control or manage their symptoms. It may work for short periods of time, but there's often relapse in these conditions. And so the larger group, AGIRA, is interested in not just a serological and a clinical and psychological assessment of these individuals, but also looking at the microbiome, especially that associated with the small intestine, with the view that the subtypes of functional GI disorders may actually be subtyped on the basis of more quantitative or objective assessments based on these 3 areas of clinical measurement. And the ultimate goal is to come up with improved immune targeting therapies, maybe histamine blockers, for instance, as well as probiotics or version of antibiotics that may alleviate the role potentially that the microbes play in the different subtypes, ultimately leading to integrative care involving approaches that include lifestyle changes as well as diet. So my role in this activity is obviously to try and take a look at the gut microbiota, especially that community of microorganisms that resides on the mucosal tissue in the duodenum, okay, just after the stomach, okay. So my work in this space is enabled by the development by Gerald Holtman of a specialized type of biopsy forceps that we call the aseptic biopsy device. And so during routine endoscopies at the PA hospital, this sort of device is used, and basically the forceps are housed within this sheath, which is protected by a sterile glycerol plug. And so as it moves through the working channel, the forceps themselves aren't exposed to the external environment until the end of this sheath is positioned where the sample of tissue is desired to be taken from. The forceps are extruded, the tissue is collected, and then the forceps are retracted back into this sheath. Well, so what? Who cares? Why is that important? Okay, so basically what it's allowing us to do is take a sample of tissue, but protect that tissue and the microbial community that's associated with it from being contaminated by luminal contents or saliva as we go through the endoscopic procedure. And as a result, we can use those DNA sequencing techniques that I talked about before, especially 16S profiling, but we can also use quantitative PCR approaches to get a measure of bacterial load on the tissue. And of course, we can do culture. So one of the things we've been doing is using nitrogen gas in the working channel as this device is removed. We put the forceps and the entire device inside a plastic bag with a couple of pellets of dry ice, put a rubber band around it, and effectively carry that plastic balloon back over to my lab at TRI, which takes about 2 or 3 minutes, move those forceps into the anaerobic chamber and ensure that that tissue is not being exposed to any atmospheric concentrations of oxygen or air, and then undertake actual culture from those tissue samples. So what have we learned using this particular device? So using that 16S profiling approach, we can show that there is a clear differentiation between patients that are ultimately diagnosed with a functional dyspepsia compared to non-functional dyspepsia control subjects in terms of what types and kinds of microbial members make up the community that's associated on the tissue collected from the duodenum. So there does appear to be distinct changes in the types and kinds of microbes present in response to this sort of functional GI disorder. There's a range of other tests that are done within the clinical labs there at PA Hospital. One of them is to look at gastric emptying, and another interesting finding that we've made is to look at the relative abundance of a bacterium called Veillonella that's present within the tissue samples collected from these subjects, and what lag time in gastric emptying is reported by their testing. And what you can see here is there's a pretty strong negative correlation in terms of the lag time and the relative abundance of Veillonella species. And in a pilot study that we published in Gut a couple of years ago, again using this device, we were able to kind of get a sense in terms of what the bacterial load is on tissue. So the way we do that is we use qPCR-based approaches using primers that target just bacteria and get a copy number of the number of genes that we have present on that sample. And we also use qPCR to measure the number of copies of the gene encoding beta-actin. So we normalize those bacterial signals relative to the sequence content that we have of human DNA. And as you can see here on the left, after patients have a nutrient challenge and report more severe symptoms associated with that nutrient challenge, a drink, there is an increase in bacterial load on tissue, suggesting that more bacteria on the tissue in the duodenum is linked with increased symptoms in this nutrient challenge test. And likewise, these patients, before undergoing endoscopy and this sort of analysis, reported by questionnaire their quality of life associated after eating or drinking. And you can see again that their quality of life decreased, and on endoscopy, looking at these tissue samples, those with a poorer quality of life reported in the questionnaire have a greater bacterial load or burden on duodenal tissue, suggesting that it's not just important to kind of try and look at what types and kinds of organisms are present and identify a potential infectious microorganism. It's also knowing about the size of the population and the community and the strength of the signal, the volume switch, and how far it's turned up by the size or the density of the community that's associated on that tissue. So we've isolated a number of these Veillonella species. This is work I've done in collaboration with Tim Wells at the Diamantina Institute. And the isolates we have are listed here, and they fall inside this green box principally, okay? And the green box, the more green, the sequence of the genome for these isolates is more similar to each other. And as you move away towards the red, the less similar they are. And the point I'm trying to make is that they're highly similar isolates that we've recovered from multiple patients here, but their relationship compared to other isolates of Veillonella that's been recovered from the oral cavity or from stool is represented by these orange and red boxes, suggesting that the genomic content of a bacterium called Veillonella in the duodenum does possess regions of functionality that are distinct and can be used to differentiate between the strains of Veillonella in the small intestine compared to those that we find in the oral cavity or in the colon. So what's important here is the opportunity to move away from just naming things and start to build a picture in terms of the genomic variation and how that may attribute to site specificity of colonization and interaction with the host through the cell communities and the environment that's present in that particular site. So just to wrap up on that, we've shown that the diversity of duodenal mucosa is quite different and altered in functional dyspepsia patients. It's also influenced by smoking. It's also different in patients with non-celiac weak gluten sensitivity. We've got a range of papers close to publication in this area. The so-called aseptic biopsy device developed by Gerald really has opened up new opportunities for us to look at microbial load as well as isolate bacteria from the duodenum. And some of these are actually demonstrating a degree of seroreactivity in subgroups of patients categorized with different types of functional GI disorders, suggesting that there may be an opportunity to develop serological tests for the diagnosis of these conditions. Okay, I want to shift now and talk about some of our work with inflammatory bowel disease, with Xu Ning at Chinese University of Hong Kong and with Michael Kam. Just very briefly, I'm probably preaching to converted here, but Australia has one of the highest global incidences of Crohn's disease and IBD in general. What we're also finding, as I understand it as a non-clinician, is that persons that have emigrated to Australia from countries traditionally thought of being protected or not susceptible to inflammatory bowel diseases, after 1 or 2 generations, their children and their grandchildren are showing an incidence of Crohn's disease or IBD at a level that's similar to their Western counterparts in the same country, suggesting that environment and lifestyle choices is having some influence here. And of course, in parts of China and in Hong Kong in particular, there are changes in lifestyle and environment that are taking place as the countries become more developed, and the incidence rate of Crohn's disease in particular is steadily rising amongst these communities. So we're interested in looking at a multinational study funded by the Helmsley Charitable Trust to see if populations here in Australia, Hong Kong and China, in terms of persons diagnosed with new onset Crohn's disease compared to first-degree relatives or housemates, whether we can identify the degree of similarities or differences that might exist in terms of dietary pattern, as well as the microbial communities associated with the colonic mucosa, not stool, but the colonic mucosa of these individuals, that might be deterministic or predictive of disease development and onset. So one of the things that I was challenged with by Michael in particular was, Mark, I'm tired of working with stool. I don't think it's as relevant as it really should be. I want to be able to use metagenomic sequencing with tissue samples. Make it happen. And I said, yeah, okay. It sounds easier said than done. But this is the approach that we've taken, and I'll share with you some unpublished results that we soon hope to have out there that gives you a sense that I think this approach is quite exciting. So we have matching biopsies collected from 5 Crohn's disease patients by Michael's IBS services team at St Vincent's Hospital, collected 6 months post-surgery to remove tissue for severe inflammation from Crohn's disease. And we've had a biopsy sample stored in RNA later, principally for nucleic acid extraction. But we took a second biopsy and moved it into a buffer prepared under anaerobic conditions and with glycerol added, so that we could put this in the -80 freezer, and that tissue would be cryopreserved so that we could extract it out and use it to inoculate microbial culture of habitat simulating medium. And so our intent was to use DNA extracted from this tissue and undergo some DNA-based enrichment protocols designed to remove as much host DNA as possible and increase the amount of microbial DNA that's present from that biopsy sample, and then subject it to high throughput next generation sequencing. And in parallel with a matching biopsy, produce a culture of the microbes that are associated with that tissue, extract the DNA from that microbial community, and then sequence it as well. And so what we're trying to do here is bypass the situation where the bulk of the DNA being subjected to sequencing is human in origin. Instead, it's from the microbial community itself. And then we take that and we can subtract away what reads are present within the metagenomic data sets that match with the human genome. And then we can work through an assessment in terms of a profile, in terms of what types and kinds of microbes are present. And we can also use other bioinformatic platforms that look at the metagenomic data in totality and give us a sense of what functional categories are associated with these communities. So for the first time, start to build a picture not just of what types and kinds of bacteria are present, but also what functional attributes they have and how these communities may differ from what we see in stool and may be relevant to the severity or the site or the recurrence of inflammation in these Crohn's disease patients. So our first step was to look at how well we use the subtractive enrichment procedure. It seemed to work reasonably well. So in other words, compared to the total DNA extracted from the biopsy, if we took a subsample of that and tried to use a particular technique that tries to strip out as much human DNA as possible and then use various methods to try and quantify the amount change in terms of microbial signal, we could get somewhere between a 2 and an 8-fold increase in bacterial signal. So we were using the techniques okay, and these sorts of data are fairly standard for what's achieved with these types of samples. Over here on the right, I show the DNA profiles of the communities as predicted from the biopsy or from the DNA sample that's been enriched, okay, to what we identify from the microbial culture, what I call the outgrowth. So just in reference again, this is tissue that's been taken into culture media. The microorganisms that are still viable there can grow. We produce a community of microorganisms, a consortia. We harvest that, extract the DNA, and we effectively have microbial DNA to work with. And in the initial instance, we've used 16S profiling to look at these communities. Don't get hung up on these particular terms, other than this is essentially looking at how similar or different the communities are with respect to presence-absence data, whether a bacterial signal for that particular group of microorganism is there or not. And these axes are a representation of how much of the variance between the samples is explained by those two axes, and the distance that exists between samples is an indication of how similar or dissimilar they are to each other. The further away they are from each other, the less similarity they share. Okay, so hopefully I've explained that effectively, but basically each colour represents a different patient, and you can see that the samples appear to cluster effectively with respect to individual patient. And in the great majority of cases, what we're finding is that the profile, the microbial community that would be predicted from the outgrowth sample matches very closely with the PCR amplicon libraries and the profiles we produce from biopsy DNA or biopsy DNA that's been enriched for the microbial fraction. And the last point I want to make is that the retention of the microbial communities in culture with the individual samples, as opposed to these all congregating in one particular spot. So again, so what, who cares? Okay, so what this basically means is that by culture, we are capturing a good representation of the microbial community, okay? Second of all, it's staying resident or most closely affiliated with the data sets that we can produce from DNA from tissue. So we're not getting any kind of bias introduced by the culture method in terms of the communities that we have. In other words, the profiles are patient rather than method specific. If the outgrowth cultures using the same medium resulted in just the same 3 or 4 different bacterial species growing from the sample, you'd expect all of these diamonds to cluster together in a particular point, and that hasn't happened here. So these data as a first pass assessment of the community suggested that we were doing really pretty well in terms of capturing the community. And now I've shifted here, so what I showed you a moment ago were the 16S based profiles. Now I'm showing you the metagenomic data, the prediction of the types and kinds of microbes present based on the functional attributes of the community produced from that type of DNA sequencing. And the red boxes represent the metagenomic data and the profile predicted from total biopsy DNA. The yellow boxes are the enriched DNA from biopsy tissue, and the green boxes represent the taxonomic diversity that's captured in the metagenomic data using the microbial cultivation as the first step. And you can see that the representation of the data or the microbes in the shotgun data produced from the tissue biopsy or the microbial enriched DNA from the tissue biopsy are well represented within our microbial cultures, but you can see a much greater expansion of data that relates to the microbial community in the outgrowth cultures compared to trying to predict community structure and function using total DNA extracted from the tissue sample itself. So again, what we've done here is expanded our representation and functional understanding of the microbial community associated with the tissue in these 5 samples collected from 5 different patients, Crohn's disease patients. And we've been able to assemble some of that data into individual genome sequences, and these are referred to as metagenome assembled genomes. So in other words, you take the quantity of data that's being produced and you stitch these small pieces together that are predicted to come from the same taxonomic group, and sometimes you can almost build a complete new genome representing that bacterium even though you haven't necessarily cultured it. Okay, so it's like an assembly of the genome for these different bacteria. And so we see a fairly large number and great variety of diversity that's represented in that data set. But importantly, the other thing that we've got to go back with beyond this step of DNA sequencing is the consortium of microorganisms that we cultured in the first place. And so based on these data, we can go and search for specific bacterial species or microbial species that we think are relevant and important in terms of host microbe interactions associated with inflammation and Crohn's disease. And so when we look at these data using the functional assessment, we identified that the representation of enzymes involved with urea breakdown are really quite high in these particular communities. So we're able to go back to the microbial cultures, and we use selective strategies to isolate bacteria that produce urease activity and identify and isolate specific organisms that possess that sort of urease activity. And we approach this in part because of the enrichment of urease genes that we could see in the metagenomic data sets, but we also were aware of a published report from Gary Wu's group that had recently shown that ammonia derived from urea appeared to be a metabolite signal or biomarker of active inflammation in Crohn's disease, and they constructed a recombinant E. coli strain that introduced the urease catalytic abilities to that strain, and in a mouse model were able to show that inflammation in the presence of that urease positive recombinant E. coli strain was more severe as a result compared to the parent strain that was effectively urease negative. So our use of this microbial culture metagenomic sequencing, I think, provides us real opportunity to have a much closer examination of the mucosa-associated microbiota and its impacts on host response. So just to wrap up this third part of the talk, it removes contaminant host DNA, allows us to use the next generation technologies. We can compare genomes between mucosa-associated and stool bacteria, start to identify how the communities may differ with respect to the site of inflammation and its severity. We're recovering low abundance taxa that are not readily detected by PCR-based amplicon sequencing. And of course, and most importantly for a microbiologist like me, we now have extant or existential forms of these microbial communities. And so rather than trying to make predictions by association, we move to an era where we can actually do some hypothesis-driven research using ex vivo tissue samples or animal models to try and identify and dissect cause versus consequence and the role of the microbiota in these conditions. Shall I stop here? I know this will only take about 5 minutes, but shall I do it? Okay, all right, okay, thank you. All right, so that's the basic science. I hope you stayed with me. I hope it was interesting. But I want to talk a little bit about how you can make friends with salad, and the gut microbiome has a role to play in that. Okay, so how can you do it? It's through food choices and food preparations that salad can make an impact upon the function and well-being of your gastrointestinal tract. So first of all, try potato salad. Raw potato starch is often a high content of what's referred to as amylopectin or amylose, and during the cooking process in water, when you boil that potato, the starch swells, the polysaccharides are pushed apart, or the starch swells and water gets in there, but as you cool that starch down and the water is repelled out of that chemistry, they stack together. The polysaccharides stack together much more tightly, and structurally, architecturally, that is more resistant to enzymatic attack, especially by the amylases that exist in our mouth and small intestine. So basically, cooked and cooled starch whether it be potatoes or rice. So if you like eating cold rice, sushi or something like that, you have retrograded starch, which is more resistant to small intestinal digestion and absorption. And this starch reaches the large bowel and it feeds the hungry microbiome that resides there, okay? And so many of these microorganisms, which appear to be in high abundance, especially in healthy subjects, but are often depleted in patients suffering from an inflammatory, chronic inflammation like obesity or Crohn's disease or some of these other conditions, they like starch and they produce a variety of goods and services that have positive attributes in terms of maintaining the structural and protective elements of the gut mucosa, and they provide a variety of different nutrients, vitamins in particular, but butyric acid, especially in terms of providing a fuel and an immunomodulatory function that helps to promote gut homeostasis. So feeding your bugs resistant starch is one way to make friends with salad. So if you like potato salad, make sure you have some at lunchtime today or on the weekend with your barbecue, okay? But don't forget about the FODMAPs. And what FODMAPs refers to are fermentable. Okay, this is a specific term relating to the bacteria of the large bowel. And it's the oligosaccharides, disaccharides, monosaccharides, and polyols that naturally occur in foods, which are, again, they escape digestion within the small intestine, but they are fermentable. They can be used by the gut microbiota to support their growth. And the work by the Monash group in particular, Jane Muir and Peter Gibson, has really identified and moved forward the concept of using diets low in FODMAPs as an induction therapy for symptoms of patients diagnosed with irritable bowel syndrome. So this paper was published 4 or 5 years ago in Gastroenterology. It was the first placebo-controlled, double-blinded, randomised controlled trial looking at low FODMAP diets, and statistically showed that there was a significant decrease in symptoms in patients with IBS, but there were no variations in these parameters or measures in persons not diagnosed with IBS. So it was an IBS-specific response. They've gone further to suggest that low FODMAP diet is also a corrective or relieves symptoms in persons that report gluten sensitivity and are non-celiacs. But the pause that we need to take in relation to these sorts of diets is that these are fermentable carbohydrates, and in another period of time in our life, especially in young children, a number of these so-called prebiotics are considered to be capable of supporting the development of beneficial microorganisms like bifidobacteria. And so there has been an identification that luminal bifidobacteria are reduced by low FODMAP diets in addition to some other microbial communities. And so there is suggestions and recommendations from nutritionists and dietitians, especially from Monash, that your adherence to this diet should be kept to a restricted period of time with the view of trying to liberalise your diet and reintroduce foods, because we still don't really understand the long-term consequences of this type of diet on your gut bacteria. So what are some foods with low FODMAPs? A variety of different fruits and vegetables are listed here. Depends on the ripeness of the banana. Bright yellow bananas have low FODMAPs. Black bananas have high FODMAPs, okay, because the starch starts to break down to fermentable sugars. Foods with high FODMAPs, well, there's probably a good number of persons in the audience that might identify one or two of these particular types of foods, and based on experience will say, if I eat a lot of that, I kind of feel a little bit uneasy, okay? So, it can have impacts, okay? So the point I'm trying to make is, yes, you can make friends with salad. Salads can be a good source of different forms of fibre, including resistant starches, and they feed you good bugs. And if you want to see a really nice video in terms of how this might work, at a more biological level. There's a video from CSIRO called The Hungry Microbiome that you can watch. But you need to listen to how your body responds to the foods you eat. You can also have too much of a good thing, including beer, okay? So beer could be considered, depending on what volume of it you drink, it could be considered high FODMAP. And the app that's been produced by the Monash Group, many of you I'm sure are aware of it, but it's highly valuable and very popular. So, just to wrap up, who thought you'd spend so much time thinking about the contents of this truck? But a number of ways we still really don't know very much about it or what's around the corner in terms of the research. This is a big problem. A lot of it's still associations. Bradford principles are helpful, but we still really need to work out what comes first, the chicken or the egg. And watch the public media and the web on this, okay? There's a good number of activities out there online that are willing to treat people like this mushroom, okay? But as a microbiologist that has no medical qualifications but enjoys working with clinicians, at least from my perspective, I think the gut microbiota do have great opportunities to provide new ways to treat and manage chronic non-communicable diseases. So I believe in innovation, as Bill Gates said, but to get that, you fund research and you need to learn the basic facts first, okay? So in this space, I still think we've got a hell of a long way to go before any sorts of claims can be made or evidence-based recommendations are developed, but you've got to get started. And with that, I just want to acknowledge the wonderful collaborators I have in clinical gastroenterology at Queensland, at Newcastle and Sydney, in Melbourne, Michael Kam's group, and in Hong Kong, also at Monash University with Jane and Peter, and the basic scientists at the Australian Centre for Ecogenomics and other collaborators at the University of Queensland. And thank you very much for your attention.
Thank you, Mark, for that fascinating talk. It's certainly a hot topic, the gut microbiome, and it appears that it's not just what we feed ourselves, it's what we feed our microbiome that's important for health. So I'd like to take any questions, if there are any from the audience. Yep. We'll just wait for the micro. Yep.
Hello, I'm a midwife, so I was just wondering if you had any comments to make about vaginal birth versus caesarean birth and things that we know about that in relation to the microbiome.
Yeah,
look,
it's there's a long-winded way of answering that question, but I'll keep it short. Okay, so especially in the first few days postpartum, the microbial communities are very different in between natural delivery and C-section deliveries, okay? The oral microbiome appears to be greatly different, okay? An antibiotic used by the mother during the final stages of pregnancy also can have an impact. And so when you have C-sections and a prophylactic dose of Keflex or something like that, it can carry over influences, okay? There is an interest now in terms of trying to develop a clinical equivalent to fecal microbiota transplantation, where a vaginal microbiota transplantation is used with infants born by C-section. I think it's still in its exploratory stages, but the reality is that there are differences, and some people would predict that these differences are imprinted longer term into the child's development by being born through caesarean section compared to a natural vaginal delivery. I think the key point of it, just to finish off, is that when we talk about fecal microbiota transplantation, everybody has one that is delivered naturally by a vaginal delivery, okay? You will be exposed to the stool microbiota of the mother during vaginal delivery, and that step is essentially removed when you have a C-section.
Any other questions?
I'm a registered nurse. My name is Sajeev. I've been reading about microbiota in relation to mental health conditions. So I feel that fecal microbiota transplant has a very good potential in treating mental health conditions instead of the monoamine hypothesis that we actually base at this time. It's based on the fact that when the monoamine hypothesis was first formulated or put forward, we were not aware of the fact that dopamine was produced peripherally, and serotonin also was produced peripherally. So now we know that 95% of serotonin is produced in the gut, 46% of dopamine is in the gut, and then we also know that the microbiome has the potential to, make these the precursors of these neurotransmitters to cross the blood-brain barrier. So when we try to address the imbalances of monoamines in the brain by way of giving chemicals, why not we treat these people with fecal microbiota transplant?
Yeah, okay. So I think to go to the heart of the last part of the question, why we aren't, is because we still just don't have the evidence base there of demonstrable impact via that intervention. So I think theoretically this whole idea around what are being called psychobiotics, the idea that particular strains of bacteria might be used as probiotics, but they're goods and services, their products during growth within the intestinal tract can have beneficial effects through the gut-brain axis on anxiety and depression, I think still being examined, and some of the initial work done in Ireland with mouse models was really quite promising, but when that was moved actually to a clinical study, it fell apart, okay? So this maintains, as I understand it, a fairly long-standing history where interventions through probiotics in particular show great promise in mouse studies and pilot studies with small numbers of subjects, but then when they're moved into something that is much more real world in scale, it starts to break down, okay? Some people respond and others don't. So where am I going with this? I think what we haven't done in the past in a lot of different areas of medical research, pharmaceutical development, is we don't take enough information or metadata, clinically or otherwise, of persons entering into the studies, so that after they've gone through the intervention and are unblinded, you've got some sense in terms of how you may be able to stratify patient response compared to what they were like when they first entered into the study. So that's my way of saying that I think that these probiotic-based interventions have considerable promise, but only in a small number of groups of people. And there's a need for us to identify what groups they may work more effectively in and use that as an intervention that's more predictive and therefore personalised rather than just this blanket approach like is currently used with functional GI disorders, where it may provide some temporary relief or no relief at all. And it's because we still don't understand who might be a responder or a non-responder to that intervention. Yeah. So we've got a long way. This is what I mean by it's easy for people to get out there and put all this hype out and on the internet provide great promises of impacts, but the reality is humans are humans, and we've still got to work through that sort of relationship better first.
Any other questions? No? I had one, Mark. You spoke about the second generation immigrants from countries with a low prevalence of, say, IBD, now having a similar incidence of these sorts of diseases. Is that due to the hygiene hypothesis, do you think, or is it a loss of gut microbes that are, functional in our guts? Mm-hmm. Can you comment on that?
Yeah, look, I think 5 to 10 years ago, the guilt by association aspects would have been what's happened in Western countries to a certain extent, that may be adopted by the persons that move there that are contributing to this. So antibiotic use, the so-called hygiene hypothesis, to a certain extent, living in a built environment are all kind of, and diet are all considered to be contributing factors. And that, to a certain extent, I think, has now been demonstrated to have caused a shrinkage of the gut microbiota in particular in individuals in those particular societies compared to what may have existed elsewhere in an urban, non-built, rural sort of environment where there were a lot more animals and animal contact and things like that. So there is a suggestion that even in vegans in Western countries, their population does not develop the level of diversity that you might see in vegetarians in well-nourished regions of Africa and the like. And it's because some people propose that these bacterial species are now extinct in Western communities. Whether they have a role to play in terms of providing protective effects or not, I think is still debatable. And probably one good way of generating that conversation is the rate and the incidence and prevalence of bacterial infectious disease in those communities in non-Western countries compared to Western populations. So the incidence and prevalence of infectious disease is still very high there, and sure, there are missing microbes between the two, and it's interesting and an interesting hypothesis to think that some of those may be protective, but the reality is antibiotic use in those communities is still pretty rampant, and epidemic forms of infectious disease are still not uncommon in those parts of the world. So they're at least not protecting against those sorts of situations or circumstances. So we've got, yeah, there's more to do.
Great. Are there any more questions? If not, we might thank Professor Morrison for his talk today. It's been great having you here, and
my pleasure.
Sure
Thank you very much.
Enjoy your beer and feed our microbes
later
this evening.
Yes, I'm looking forward to some FODMAPs.
Yep, great. Okay, put our hands together.
Thank you. Thank you, Claire. Thank you. Thank you very much. Right.
Well, that concludes everything for Charm this year. I guess we'll see you next year. Or maybe not, depends how I feel. Thanks, everybody, for your contributions.